Sorry, just an obvious reminder, but if you could put your phones on silent, that would be fantastic. Thank you.
Okay, morning everyone. It's 10 past, so I'll crack on and get through the introductory bit so we can move on to the interesting science part. So welcome to a huge number of people in this room. It's fantastic to see so many people back in a conference again. There's something like 230 almost of you attending this meeting in person from 22 countries, which is absolutely fantastic after 2 years of postponed meetings and all the stuff that we all know about going on. The upside of having to adopt all that virtual technology for the last 2 years is that we're also joined by 350 odd people online, probably more than that now, from 40 countries, and I'm absolutely delighted that we have that kind of global reach for animal welfare science now. So welcome to everyone who's hopefully watching virtually online as well. It's a real pleasure to have you here too. I've got lots of boring practicalities to tell you before we get on to the interesting stuff of the day. If there's a fire alarm, there's no practice fire alarms planned, so please leave the building. If you turn left as you come out of the building and assemble outside 14 Queen Street, which I think is the British Medical Association, we can check that everyone's out of the building. In terms of catering, we'll be doing tea and coffee and lunch in the Great Hall and the library upstairs, and also in the quiet area around the back of this lecture hall. So plenty of different places to get your tea and coffee and lunch. The food is vegetarian with vegan options. That's not a policy stance from UFAW so much as the fact that we really struggled to get welfare assured meat from our caterers. So we took the decision to stick with vegetarian and vegan because we can assure you that at least the eggs and the dairy are RSPCA assured, and it's important to us that we can tell you what welfare standards the food we serve was raised to. If you had very specific dietary requirements above and beyond vegetarian or vegan, and you let us know about it, then we should have food prepared for you. Just speak to the catering staff and they can help you out with that. Tonight we're going to have a drinks reception in the Great Hall, again upstairs at the end of the meeting until about 8 o'clock, with some nibbles hopefully, and then you can head off into town and enjoy Edinburgh for the rest of the evening, hopefully, if you've got more energy than I have. Really important one, there's the Wi-Fi code if you need it, free Wi-Fi. It'll be on those other screens outside as well, hopefully. And that hashtag in the bottom, UFAW 2022, if you want to use the Wi-Fi to tweet about the meeting, that would be great. Really boring bits, we will be taking photo and obviously video because we're live streaming this conference, so just be aware that we are filming. If you need an attendance certificate, you can get one from the folks on the registration desk. As well as all the great talks today, we've got poster presentations. There's something nearly 80 odd posters upstairs. The presenters should be with their posters during lunchtime to explain them to you. And there'll be a competition for the best poster, which we'll announce at the end of the meeting. Folks who've got posters, could you take them down before 5 o'clock tomorrow, because we've got another meeting to set up in here. Ideally, perhaps before the workshop tomorrow afternoon. And just to explain for those in the room, this doesn't affect the folk online. Tomorrow afternoon, there's a debate forum or a workshop for you to attend. You should have told us already which one you want to go to, so please go to the one that you booked. If there's a little black dot on your badge, that means you're going to the debate about animal welfare labelling. Otherwise, you'll be staying in this lecture theatre to do the workshop on measuring behaviour better with Melissa Bateson. So the debate forum will be upstairs in the meeting rooms number 1 and 2. Okay, just a little bit of plugging for UFAW before we move on to the science proper. Lots of you probably know we publish a series of animal welfare books with Wiley Blackwell. Lots of really great animal welfare texts. They give us a code for people who've registered for this meeting. So if you'd like to get these books with 20% off, just go to wiley.com and use VET20 and you can have a discount. And finally, I need to do a bit of promo for UFAW itself. For those of you who don't know much about us, I always like to explain what UFAW actually is, because I think the name maybe doesn't do us any favours. We're not beholden to any universities or any government. Despite the name, we're a charity. We're an international scientific and educational charity, and it's our mission to try and help discover what matters to animals, develop solutions to animal welfare problems, and disseminate the evidence through things like this meeting. And the only way we can do that is if people give us money to help us do it. We're supported entirely by donations and legacies and people paying their membership fees to us. So if you'd be interested in becoming a member or helping us support meetings like this, that would be greatly appreciated. There are links there, especially for those people who are coming online and attending this for free. It would be really helpful to have some support to keep these meetings free for people around the world who struggle to have the funding to attend them. So thank you, and thank you to everyone who already has donated. A couple of upcoming events. I know we haven't started this one properly yet, but a conference we're jointly supporting with the UK Centre for Animal Law, celebrating 200 years of Martin's Act, the first animal welfare law, by looking at animal welfare law visions for the future, is coming up. So have a look at that if that one's of interest. And because we're very conscious that our friends on the other side of the world, in Australasia and Southeast Asia, are often attending these conferences virtually at appalling hours of the middle of the night, the next event we're trying to organise is an online conference on their time zone. So organised out of Australia by Mia Cobb and some colleagues in the region on the 6th of 8th of December online, and we'll be putting out a call for abstracts for that one as soon as we can once we've finished this meeting. So save the date for that one as well, especially if you're in that part of the world, because we're keen that they actually get to do a meeting at a civilized time of day for once. And with that, all I need to do is say thanks to the folk who made this meeting possible, which is not so much me as Steve and Beata, who are over there, who are very responsible for the programme and all the organisation, Louisa and Liz, and all the folk back at the office. All of you for coming. It's fantastic to see so many of you actually attending in person, and everyone who donated to help make the online meeting possible. So with that, we can get on with the science, and I'm delighted to introduce Dr. Katie Cronin from Lincoln Park Zoo as the first session chair, great friend of UFAW, and going to be a fantastic session chair for the first session. Thank you all.
Good morning, everyone. I'm Katie Cronin. I'm from Lincoln Park Zoo. I'm the director of the Animal Welfare Science Program there in Chicago, in the States. And I am pleased to be able to chair the first session. I have a couple notes to share. Speakers have been asked to leave 5 minutes at the end of their talk for questions. If you have a question, we ask that you make your way to one of the several microphones that are along the side, up the middle, the stationary microphones there. Unless you would like someone to bring you a microphone if you have a question, we can also do that. We have a roving mic. And if you make your way to the middle or to a microphone or raise your hand, then wait to be called on for a question. If you do ask a question, we ask that you introduce yourself and tell us where you're from before you get started. So with that, I think we can introduce the first speaker. I'm very pleased to introduce Professor Hanno Würbel from the University of Bern in Switzerland, who will be talking about how to improve the quality of animal welfare science. So, thank you.
Thank you very much, Dr. Cronin. Thank you for inviting me, and UFAW, and welcome everybody. It's so cool to be back in a meeting with real people and not just talk to your notebook all the time. So I'm really pleased to give a presentation on some of my more recent work on science, basically, meta-science related. And I've been asked to talk about quality of animal welfare science. Now, of course, it would be exaggerated if I would claim that I can assess the quality of animal welfare science. And I'm not going to do this simply because we lack the evidence. In order to assess the quality of science, we need a lot of meta-science. And when I was looking up the literature on this, I just couldn't find much on science, on animal welfare science. Of course, we all know that there are issues with the measures we're using, with the study designs we're using, the animals we're using, but there is really not much science on science in animal welfare research, as opposed to, for example, biomedical animal research. And so I'm going to draw heavily on evidence that we have from meta-research in biomedical research, and then, where possible, try to draw some links to issues that are of specific concern to animal welfare science. That was the wrong button. So what do we even mean by quality of science? It's how good or bad something is, and good and bad, of course, are value statements. So what is good science? What is good animal welfare science? Is it science that answers relevant questions? Is it science that uses rigorous methodology? Or is it science that is morally responsible? And of course, it's all of these. Only science that is relevant, rigorous, and responsible can be good science in all its facets. But who is deciding what good science is? And I think we have a division of labor here in the science system. So, for example, whether science answers relevant questions, that is something that usually funding bodies decide. And the science system trusts funding bodies because that's why allocation of funding, of third-party funding, is such an important criterion in the promotion of researchers, for example. Whether science is rigorous in its methodology, that's something that we have delegated to our peers, so we're all part of that system. Peer review is kind of the gatekeeper of science and decides whether or not something makes it into the domain of scientific evidence or not. And whether or not the research is morally responsible, that is something that ethical review boards are meant to decide. Now, I'm going to focus on rigorous methodology today, largely, and that's, of course, because that is what I'm studying and what there is most evidence around about, but also because, of course, unless methodology is rigorous, scientifically rigorous, we're not going to answer any relevant questions, or we're not going to answer these questions in a good way. And unless methodology is rigorous, science can't be responsible. Now, if we're talking about rigorous methodology and peer review, then there is this elephant in the room that has been around for a while, and this nice chap here is the one who basically got things exploding when he published a paper 17 years ago by now, where he claimed that most published research findings are false, showing that most research findings are false for most research designs and most fields. And so that's probably also something that should be of concern for us animal welfare scientists. And meanwhile, you've all noticed this, I'm sure, we're talking about a so-called reproducibility crisis. This is data from a survey conducted by the journal Nature some years ago, where they asked scientists whether they perceive the situation as being a crisis in terms of reproducibility, and 90% of the respondents of this survey answered yes, and more than 50%, more than half of the scientists who responded to the survey considered the crisis to be a significant one. So what I'm going to talk about in the next few minutes is based on things that have happened since. So basically discovering that there are issues with reproducibility, and when I'm using the term reproducibility, I really use it in a very broad sense, including scientific validity, translatability from study populations to target populations, so covering the entire space of scientific rigor. So what are potential causes of poor reproducibility in animal research, and maybe more specifically in animal welfare science? Of course, when working with animals, we have to start with the animal model. The animal model being a model organism on the one hand, but it's also an intervention usually that is done on animals, and an outcome variable being measured on these animals. And so one of the ways in which animal models can go wrong is that they do not properly translate to the target population that we have in mind. And of course, that is one of the main issues in biomedical research, where the majority of research is conducted on mice, on laboratory mice. And of course, that's always an issue, is how translatable can research on mice be for human beings. And more recently, it wasn't just about the difference in species, so the species differences between laboratory rodents and humans, but also the fact that the way in which we treat these animals and house these animals is so fundamentally different to the natural world that we are exposed to, that people are now starting to rewild these animals in order to get closer to a translatable model. And a particular example are the so-called wildling mice, where laboratory mice or strains that are used as laboratory animals are by embryo transfer brought into wild mice in order to catch their natural microbiome and then breed these wildling mice that seem to be a better model for a number of disease states in humans, especially everything to do with immunology or processes that are dependent on the immune status of animals. Another way in which animal models can be wrong or inappropriate is if they are housed or cared for in a way that they do not develop normally, that they develop abnormally. And this is a picture from my very first study in applied ethology, where I was looking at housing conditions of laboratory mice, finding that more than half of all laboratory mice would develop abnormal stereotypic behaviors under the conventional housing conditions. And at that time, that was, as far as I remember, one of the first times where a welfare issue wasn't just treated as a welfare issue, but also raised concerns in terms of the scientific validity of the results obtained when using these animals for biomedical research. And then, of course, there is what we measure about animals, and there's a huge literature about the scientific validity and the different dimensions of validity of measures of diseases, and in our field, measures of animal welfare. And this is just one example to highlight the problems. The so-called forced swim test has been around for a long time in biomedical research as a test system by which, on the one hand, states of depression are induced in mice and rats, and on the other hand, substances are measured for their efficacy to treat states of depression. Now, researchers studying this test and the construct validity of this test have long argued and demonstrated that this test essentially lacks any construct validity for states of depression. And yet, researchers have continued to use it for that particular purpose, for others as well, but also for that purpose. And of course, that's not just bad science, but it also makes them an easy target to animal rights groups who can then take this as an example and demonstrating that the scientists don't even trust their own evidence and abuse or misuse tests for no good reason. Now, do we have issues with model validity in animal welfare science? One of the good things is certainly that most of our study populations are often a lot more representative of the target populations that we're aiming at. So we're not translating from mice to humans, we're usually translating from cattle to cattle and from laying hens to laying hens, because that's what we're interested in. And so both study populations as well as the study settings under which we study the animals are often a lot more representative of the target populations and target conditions. And so there is certainly a much smaller translational gap between study populations and target populations. But we certainly do have issues with outcome variables, and our measures of animal welfare, for example, they have kept us busy since that discipline exists. And this is a very recent example by Liz Paul and colleagues who studied how or whether conditions that are preferred by animals, in this case hens, whether conditions that are preferred by animals or non-preferred, whether that's also reflected in terms of other measures of animal welfare, such as judgment biases and other candidate welfare measures, such as heart rate or other more conventional welfare measures. And they found that overall the different approaches did not converge to identify a precise state of animal welfare. And from this, they conclude that further work is needed to establish which alternative measures of effective state might be more appropriate indicators of animal welfare. Now, that's a rather pessimistic conclusion, that apparently we're all using different types of welfare measures, but they don't seem to measure the same thing. Now, of course, one thing is that they're all wrong and they don't measure anything to do with animal welfare, and I would certainly not subscribe to that. But it certainly seems that they measure at least different things or measure things in different ways so that they don't simply converge and correlate with each other. And that, of course, is then a problem when we want to translate our evidence or our findings into practice and have an impact in practice, because if we cannot demonstrate that our findings are based on solid science and are replicable and translatable, then we may have a problem. But there's also another issue, and maybe that's contributing to this finding, is that animal welfare scientists, for good reasons, I think, they tend to shy away from treating animals badly. And so you can look at the literature whenever scientists in animal welfare science are probing measures of welfare or testing welfare measures, they're usually using rather subtle treatments. So they don't really want to stress the animals badly. They don't want to impose a lot of pain on their animals. And one of the problems is that, of course, with this, the effect sizes that we're able to measure, they are shrinking, and sometimes shrinking to almost immeasurable effect sizes. I'm not arguing that this was the case in this study, but if you look at what was compared in this study, it was a preferred condition versus a non-preferred condition. And of course, there was a proper nest and there was a dust bath in the preferred condition, in addition to the animals having twice as much space as in the non-preferred condition. But even this is vastly different from the reality that these animals are facing under conventional housing conditions. And this isn't even the worst conditions that these animals are facing. So the question is at what spectrum are we actually probing these welfare measures? And of course, with this, the question is also, well, are these measures really measuring? Are they continuous measures of different welfare states, or are they perhaps measures that only start to measure something above a certain threshold of, let's say, pain or poor welfare? And a lot of this we simply don't know, because in many cases it's exactly designs like these where there are two conditions that are being compared. To be fair, in this case it was three conditions, but in many cases it's two conditions that are compared, and we don't really know, even if we find a difference, whether that's because one condition is above a certain threshold, or because the measure is actually measuring a continuous difference in welfare states. So we certainly have issues with outcome variables that are often poorly validated, and we may have issues with interventions that may often be too subtle to measure meaningful effects. Now, I'm not going to go more into the issue of construct validity and model validity, because tomorrow we're going to have a keynote session on uses and misuses of measures of welfare, where this is certainly going to be discussed in more depth. So what other problems of reproducibility or scientific rigor do we have? Of course, one of the biggest issues is the use of small sample sizes, leading to a lack of statistical power. And there has been a lot of research on this in the biomedical field. And these are data drawn from a recent systematic review where they were looking at animal research in the neurosciences and in metabolism research. And the picture is the same across the board. As you can see here, listed the number of experiments and the power achieved in these experiments based on meta-analytic effect size estimates. And from this, you will find that the median power achieved by these experiments is about 18%. While what we're normally aiming for is 80 to 90% power. And even though power is not the only, or maybe not even the best measure to use for this, 18% is certainly far off that target power that we're aiming for. Now, these data have to be taken with a grain of salt, because as you can see, even null effects have been included in this meta-analysis, which is not very clever, because of course, in order to power a study to detect an effect size of 0, you have to have an infinitely large study population. But still, what you can see here is when you look at the number of experiments and how many animals they have been using, you can see that the biggest peak, the median of all studies is 10 animals per group. And 10 animals per group suspiciously sounds not like having done a proper power calculation, but probably just chosen 10 animals because it's a convenient number of animals and something that people have always been doing. So why not? So is sample size an issue in animal welfare science? I certainly do believe it is, for various reasons. One of the reasons is that often, because we're working with large animals in expensive facilities, sometimes we just don't have more animals available for studies, and then we compromise. But as you can see, it also seems to be a deeper issue in general, and that is from a recent systematic review on the use of judgment bias tests in animals across all sorts of different species. And above here you can see the overall meta-analytic mean effect size that was detected across all of those studies. And this is a standardized effect size of 0.4. 0.4 is like a medium effect size. It's not a very small effect size, it's a medium effect size, but as you can see, it is barely different from 0 because the confidence interval starts at 0.03. So maybe that is a reflection of what I've said earlier, that we're usually studying subtle interventions that don't lead to very large effect sizes. But the problem we have with this is that, of course, any of those, and there are across the different species, you can see most of them are based on a single study, others on several studies, but even those that are based on single studies, none of them was sufficiently powered to detect an effect on their own. And in order to find significant effect sizes with such an effect size, the authors reported that at least 50 animals per group would be needed to detect such moderate effect sizes. And that is not what the average effect size of our studies is like. Next, there is a lot of analytical flexibility, and I'm sure you all know this, that much of the research that we're doing is not strictly confirmatory in the sense that we set out, we define a hypothesis a priori and set up an analysis plan a priori. But we have an idea, we have a question, and then we start collecting data, and often we collect data on a whole range of outcome variables, and we may be able to subdivide our study population into subgroups of different age, of different sex, of whatever. And by this, of course, we're opening up that forking path of analysis that we can conduct and just assess a lot of sub-hypotheses as if we had been doing proper hypothesis tests. And this is what is called p-hacking. So that's one problem, in particular if we are not correcting for multiple testing, and that's depicted on this picture here, where you can see the proportion of committing at least one type 1 error, so finding a false positive just by chance, with the number of statistical tests you're performing, and even with only about 15 such comparisons, there must be at least one, or there would be at least one type 1 error in 50% of the cases. So the probability of finding chance findings increases massively with the number of tests being conducted. Another problem is harking, standing for hypothesizing after the results are known, adjusting the hypothesis to the results that you found, and of course, both of these things are going to distort the scientific evidence in the primary literature, and this is then a major problem for those who are synthesizing evidence based on systematic reviews and meta-analysis. And we have recently looked into the issue of harking based on a systematic review that we conducted on the use of behavioral tests for the study of drug development. And so we did some kind of reverse translation using drugs and anxiolytic drugs prescribed for humans, and then checked how they worked in mice in behavioral tests that are supposed to measure anxiety. And then we discovered some interesting data, and this is data from this set of data, which is data on the effect of diazepam, that is an anxiolytic but also a sedative, on locomotion in the open field test in mice. Now, the open field test is the most popular behavioral test, but locomotion in the open field test has long been known to be a poor measure of anxiety. It's often used by many researchers as a control variable to assess activity of their animals, and use that as a control variable for the more specific measures of anxiety that are based on exploration in the center of the open field, as opposed to the areas in the corners and at the border of the field. But what you can see here is a distribution. Here are the real studies, a distribution of the effects found when testing for locomotion in the open field test. And this is what the authors reported about these findings. So, of course, those who found that diazepam led to an increase in locomotion, they interpreted this as an anxiolytic effect of diazepam, leading the animals became more brave and then explored more. While those who found that diazepam led to a decrease in locomotion, they interpreted this mostly, there are some interesting counterexamples, but mostly they interpreted this as a sedative or depressive effect of diazepam. Now, there's not much wrong with this, except that these are post hoc explanations of what has been found. The concerning bit is this one, and this is the effect of diazepam as predicted in the study. So predicted means that either in the introduction or in the method section, these authors predicted diazepam to either have an anxiolytic effect or a sedative effect, or they wouldn't report anything because they just recorded it as a control variable. As you can see, those who did not make a prediction, they did not find an effect. So this is really sitting right on top of the null effect. But those who did predict an anxiolytic effect, they did seem to find one, while those who predicted a sedative effect, they did find a sedative effect. And now I really don't know how else to explain this than by adjusting the hypothesis with hindsight once you've seen the results, because otherwise that would be miraculous predictive validity of these hypotheses. So then there are, of course, many other types of biases, risks of biases that we all know of: randomization, blinding, defining inclusion and exclusion criteria a priori rather than afterwards, defining a primary outcome variable, defining a statistical analysis plan a priori rather than with hindsight. So these are all things that our undergraduate students learn in their first courses on study design and study method, and yet systematic reviews have shown that we don't really adhere to these practices. At least we do not report on them, because a lot of systematic reviews have found that when looking for whether or not the researchers would report on all of these measures to prevent risks of bias, they found that they hardly ever find statements about blinding, randomization, sample size calculation in the method sections of the papers. And this is not just bad because we then don't know whether or not there is bias or risk of bias. And many scientists have actually argued that, well, we're just not reporting, but of course we're adhering to these principles. But then evidence spoke a different language, because whenever comparisons were made between papers where these measures against risk of bias were reported versus papers where these measures were not reported, there were systematic differences in the effect sizes that were found. And in those studies, no surprise in those studies that did not report these measures against risks of bias. They generally found much larger treatment effects than those that did report these measures against risks of bias. That's why we have guidelines now, the PREPARE guidelines that help researchers design their studies and think about everything that they need to think about when preparing and designing studies, as well as the ARRIVE guidelines that are reporting guidelines, but of course by reminding scientists of what they have to report in their manuscripts, in their papers, it also reminds them of what they have to think about when designing and conducting studies. So do we have issues with risks of bias in animal welfare science? There isn't much evidence available, but this paper that is already around for some time, and I think that was pretty much the first that did look into risks of bias in applied animal behavior research, and they found that we tend to score what we believe. So if what they did, they gave their students videos to score, and they then told them that these animals were either kept under conventional housing conditions or under organic housing conditions, and no surprise, if they were expecting these animals to live under organic conditions, then the welfare assessment turned out to be much better, so their welfare state was much better than if they were told that the same animals were housed under conventional conditions, which simply reminds us that one of the problems that is very difficult for us to overcome in many cases, namely the fact that we often simply cannot blind ourselves because we see what's happening and what we want to assess, just reminds us that there is a real danger that we distort the evidence either consciously or unconsciously, just because we're affected by our expectations. And then finally, I have to mention that one, which is largely due to me, that this is on the list to causes of poor reproducibility, the issue of standardization leading to poor generalizability. And while all these other causes of poor scientific rigor and poor reproducibility are known to be causes of poor reproducibility and are considered bad things, standardization, at least in biomedical research, is actually promoted as a good thing, which is meant to increase the precision and reproducibility of results. Now, in order to understand why standardization is a problem for reproducibility, we have to go back in history when reproducibility was established as a key principle to establish scientific evidence or matters of fact, and that was sometime in the 17th century when the Royal Society decided that from now on, in order for an observation or a finding to be accepted as scientific evidence, it would have to be replicated independently by an independent, of course, by a British laboratory. Now, we've come a long way since then, and now we've replaced these independent replicate experiments by technical or biological replicates within our study designs. So instead of observing one animal, we have a study population, and we consider that population to represent our independent replicate measurements. But of course, and from looking at this, you can see that, of course, these animals within our studies, they are a lot less independent of each other than animals would be that are tested independently in independent labs by independent scientists. And especially in biomedical research, they usually share the same genotype, they've had the same food all their life, they've been treated by the same care personnel and tested by the same experimenters, so they have a huge amount of shared genotype and shared environment, and so it shouldn't come as a surprise that if someone then replicates such a study with such rigorously standardized conditions within studies, that they will find differences between studies. Now, I'm not going to go more into this. I've spent quite a lot of my scientific life working on this and publishing on this, and we've analyzed this theoretically. We've analyzed this statistically, as in this paper, and based on an international workshop, we came up with recommendations as to how best to incorporate biological variation into study designs in order to make results more externally valid and reproducible. So, is standardization an issue in animal welfare science? Well, study animals are generally more diverse in our field of research. That's certainly a good thing. And study settings are usually a lot less standardized, although some of us, of course, are also working under laboratory conditions. But in any case, and that is something that is shown across the board, is that pen, barn, farm, or study can have huge effects on the measures that we're taking, that we're using, and so need to be taken into account when designing studies and certainly when interpreting study results. And in a recent paper on this issue, Christian Navroth and Lorenz Gigax wrote that studies in farm animal welfare science can take advantage of heterogeneous farm settings that can serve as the population for sampling and accordingly follow a framework that improves external validity and thus increases replicability. And I can only subscribe to this. So these are the main causes that I consider relevant in terms of poor scientific rigor and reproducibility. And these map nicely onto the three main dimensions of scientific validity, namely construct validity, internal validity, and external validity. And because of that, and because of the 3 V's, I have coined the term 3 V's as a principle that can be applied for the assessment of study protocols for scientific validity in an ethical review, where on the one hand, we use the 3 R's principle in order to minimize harm to animals, and on the other hand, we can use the 3 V's to maximize the scientific validity and thereby positively affect the harm-benefit analysis of animal research. In Switzerland, and I was happy to see this with the last revision of the application form for animal experiments, this has been uptaken there, and now researchers need to justify their study designs in terms of the 3 dimensions of scientific validity when applying for a license. Now, I've seen the light blinking, and actually the blinking has stopped, which probably means that I should stop talking now. So I skip my 2 last slides on reminding you that it doesn't stop here, but ultimately, in order to not only do rigorous research, but also responsible research, of course, research has to be open. And one of the best ways to do this is by pre-registering your study protocols before you start conducting your studies. And then there's a lot of other issues that we should think about to make sure that our research is responsible, and only open research can be responsible research. Thank you very much. Do we still have some time?
Yes, we have 5 minutes for questions. So if anybody would like to make their way to a microphone or raise their hand, and we will pass one to you.
I'm going to steal the chair's privilege and ask a quick one. What about pseudo-replication in these kind of studies? Do you think that's a big problem?
Oh yeah, I mean there are many more questions, and some of the problems that I've highlighted, they're just examples of the problem, and there are many additional aspects that would add to these problems. And of course, pseudo-replication is a problem when basically the example I've shown you about standardization, that is in fact a case of pseudo-replication, in that we of course consider these independent animals as, and they are statistically independent entities, but of course with respect to the target population they're aiming at, they're not much more than pseudo-replicates, because they have too much shared environment and shared genotype to act as proper replicates. And I think that is often the case, and there are classical or there are like typical ways of pseudo-replication when you, I don't know, take multiple samples from the same animal and treat it as independent, as if it was taken from different animals, or when treating animals from within the same cage or pen equally independently as animals from different cages or pens. But I think we now have the statistical tools, the study design tools and the statistical tools to actually deal with this. And I would argue we can never get completely rid of pseudo-replication. I mean, pseudo-replication is on a continuum from nothing to very much. But what we can do is at least to take the hierarchy of our study design into account when analyzing the data and using, for example, cage or pen or farm as a random effect in our analysis to do justice to the data.
Oh, we have one question, if we could pass the mic.
There's a microphone, we can just move across, right next to the black one. Press the yellow button.
Sorry, I didn't expect to jump on the scene like this. Hello, my name is Pernille Fraas Jonsen, and I work for World Animal Protection in Denmark at the Danish office. And I have, maybe it's a bit provocative question, but you haven't mentioned anything about funding. I mean, how does the funding of science do you have any records of whether that influenced the results? Thank you.
Oh, yes, of course. I mean, I did mention funding. Funding is the gatekeeper of what is being studied because, of course, in order to study something, you need money, and the funders decide what they consider as relevant or irrelevant. So that's the first hurdle to be taken. And, of course, the funders, they can play a massive role in improving quality of science just by, on the one hand, by asking for certain kinds of evidence within grant applications about issues that are important for quality, but also in making sure that the reporting of the research is done in a way as it should be done, so that they really ask for proper reporting of all the studies that have been conducted. But certainly the best way, and I still think the best way of achieving a lot of the improvements that we're looking for is really by asking for pre-registration. So if researchers have to declare what they're going to do and how they're going to do it before they are doing it and before they have seen the data, then of course that's the best insurance against all of those biases that I've been talking about. And this is now made relatively easy. There are a number of platforms where you can register your study protocols in rather simple ways. And it's not just a bureaucratic procedure. Of course, it does take some time. And I think that there are some improvements possible by, for example, aligning pre-registration with applications for study licenses. But it's also something that gives you a lot in the end. When you can refer back, you present your results, and then you can refer back, look, that's how we predicted we're going to do it, and we didn't cheat and nothing. And so that is also something that by which your research and your papers gain a lot of credibility and trust. And I think that is something that you need to experience in order to enjoy it.
Thank you.
I think I should leave now.
Well, thank you very much. And with that, I'd like to introduce our next speaker, Dr. Heather Browning from the London School of Economics and Political Science. And she'll be talking about determining the line of acceptable welfare.
Yeah, thank you. Okay, so today I'm going to be talking about a research question that I've been interested in recently, and that is, how do we decide what line we use when we're thinking about what counts as good or bad animal welfare? That is, how do we pick out this baseline that counts as acceptable welfare for an animal when we're judging which conditions we should be promoting and not promoting? So to start with, I guess just a little background, the concept of welfare I'm going to be using here is this feelings-based or subjective experiencing account. I'm sure you all know there's lots of different ways of sort of defining or conceptualizing welfare. Which one we pick is going to change a little bit, what we discuss. I think for the purposes of what I talk about here, it is going to be fairly relevant no matter which welfare concept you endorse, but just so people are aware that this is what's working in the background, so some of what I talk about might be more relevant to this. I think the feelings-based account of animal welfare is a fairly common one now. There are these other accounts, physical, biological functioning, natural living, and preference satisfaction, that I think all play a role. So I think these are things that we consider to be instrumentally important in that they create the positive or negative feelings that then go into animal welfare. But for what I talk about, it is going to be these animal feelings that I take to be the important component for welfare. So when we're thinking about these thresholds or these baselines for animal welfare, I was trying to think about, when is it that they're going to be important? Because in animal welfare science, I guess there's sort of these few things that we do. So one of them is just measuring welfare. It's going out there and, taking animals in different conditions and thinking about, is their welfare good or bad under these conditions, or, does it improve or decrease? But there's also this part about making recommendations. So, within animal welfare science or animal welfare assessment, there are often recommendations made about the overall quality of some set of living conditions or some experiences of the animals. And these recommendations can take a couple of forms. So it might just be, making recommendations about what can improve welfare above the current level. So this is more a comparative recommendation. For instance, a lot of studies that say compare two different housing conditions, then find which one's better for welfare and might make a recommendation that we shift to this second type of condition so that the animal welfare is improved. But there might also be recommendations about what creates good versus bad welfare, or acceptable versus unacceptable levels of welfare. And this requires then this sort of extra value judgment on top. It's not just about when is welfare higher or lower, but it's when is welfare higher or lower than some standardised line that we take to be this line of acceptability. And this is, sometimes thought of as this, a life worth living, rather than not, when we've sort of switched past this point that welfare is bad or animals are suffering, up to the point at which we think that what we're doing is acceptable. And an example of this might be in certification schemes that are used. There are a lot of sort of labelling and certification schemes now that assess animal welfare and look at, whether particular institutions are providing an acceptable level of welfare. And here there is this value judgment in play about deciding what's acceptable and what's not. So thinking through, some of the different ways that we might set these thresholds, I've sort of thought of 3 different possibilities here that I'll go through. So the first is thinking about welfare at a zero line. Second is thinking about, what's the maximum level of welfare. And the third is thinking about welfare in comparison to some fixed reference class. So to start with the zero line, so, thinking about welfare in terms of animal feelings, the zero line then sits sort of at the middle of our scale. We've got this spectrum here from very poor welfare where an animal is suffering, through this neutral point up into good welfare where an animal's having mostly positive experiences. And that zero line then represents the shift where you move from more negative to more positive experiences, such that the pleasures of the animal, the positive experiences, outweigh the negative experiences or outweigh that suffering. And this might be a nice point because I think it gives us this sort of natural empirical ground for thinking about acceptable welfare. I think, most of our welfare measures, or some of them at least, are good at detecting valence like this. We can tell the difference between a negative and a positive valence, and we can see when this is switching across. And it gives us some reason to think that there is this, principled way of grounding welfare at this 0 point. And this is often taken to be the point at which we create a life worth living. Once an animal has more positive than negative experiences, we think, yes, here's the life worth living. I do know, though, that some people push back on this and they say, well, is it really? So, one of the problems perhaps with this view is that perhaps it's not asking for quite enough. That having, just above this 0 line where, your life is just got a tiny bit more pleasure than it has suffering may not be considered by very many people to actually be a life that they'd want for themselves or what they'd consider to be an acceptable standard of living. And if that's true, perhaps that's not enough for what we want for our animals either. So then I guess we could shift in the other direction and think about, the maximum possible welfare, the happiest animals. And here, the line of acceptable welfare would essentially be the highest possible welfare level, such that, anything below that, we're still thinking we've got some way to go to get where we need. I think there are a lot of potential issues with this view. In the first place, it's actually unclear how to set this. It's easy to say the highest possible welfare, but when you start thinking about that, you're like, well, what is the highest possible welfare for any individual? Is it the highest possible welfare they can attain sort of under natural conditions, the highest. Perhaps even if you took, a whole lot of drugs that made you really, really, really happy, is that the highest possible welfare experience? So even this, has these questions about how we set it. There's also the obvious practical issue that it's basically not at all achievable in practice. I mean, I think, even for human lives, there's going to be very, very few people in the world who ever achieve their maximum possible welfare level, because there's always going to be some amount of suffering, and we've got limited resources in which to do this. And it also creates the problem that below this line, it doesn't give us any way of distinguishing what's adequate or inadequate welfare. If we just think, look, we're always aiming for the maximum, we've got no way of saying, well, is it good enough to be at some lower point, because anything below there is just going to be bad, I guess. So I think perhaps the role for something like a maximum welfare line can be thought of as more aspirational. So my background's mainly in zoo animal welfare, and so when I think about the way we assess and ensure welfare for zoo animals, there often is this idea of, how close are we getting to a higher level of welfare? It's this sort of cycle where we're always trying to get a bit better and get closer to what could be considered some sort of theoretical maximum. So as an encouragement of aspiration or best practice, perhaps this plays a role in that sense. And finally, I think we can think about a comparison of welfare to some sort of fixed reference class. And this means choosing a target population of animals, either a real target population that exists or a hypothetical one that we'd like to see exist, that we think represents the sufficiently good life for animals, and then looking at whether or not the animals that we're measuring or assessing, whether they're roughly at the same level as this target class. Some of the problems with this might be figuring out exactly how we choose what is the right reference class. And I think that's perhaps a bigger problem that you can imagine, because there is this huge issue of being potentially arbitrary, and that we just look at some group of animals and we say, yeah, they're the ones that are good enough, everyone else should meet that standard. And I think there's potentially a strong possibility of bias here, in that we take things that we think are important or that we would want for ourselves and assume that some group of animals that has those things is the one that is right. And I think this comes out really clearly if we thought about this as a way of setting a line of acceptable welfare for humans. If we were to just pick some group of humans in the world from some country and say they're the ones that have the acceptable standard of welfare, and then just applied that to everyone around the world, we might think that there'd be biases towards which population we choose, and we may not be thinking through, what matters from the point of view of the population themselves. So, I think we have to be careful then, but there are perhaps some natural points that we can think about. So one of these fixed reference classes that might be most obvious or most intuitive is that of wild animal welfare. And so this is the idea that our fixed line is that an animal has acceptable welfare if its welfare is at least as good as that of its wild relatives, or wild ancestors in the case perhaps of some domestic animals. And I think this is a really common comparison point for people who are animal advocates. Again, coming from a background in zoos, there is this idea that, thinking about wild animal welfare, this is the life that these animals are being denied, and this is the life that we should try and encourage for them. Obviously, this is somewhat questionable for domestic animals because they have no kind of wild counterpart, a life that they're being denied, but perhaps there's some way of thinking about it that makes sense. I think there's some problems with this view, a lot of empirical problems in that we just don't actually know what the welfare level of our wild animals is yet, and there's heaps of disagreement about this. So the wild animal welfare literature has, all sorts of people going from people who say that, all wild animals live lives of fairly extreme suffering to people who think that it's quite idyllic. And obviously, we can think there are many negatives in the experiences of wild animals. So they have experiences of predation, hunger, disease, exposure to elements. But there are also positives that they experience that perhaps are hard to replicate in captive animals, which are, particular kinds of extensive behavioural freedoms and opportunities to perform different behaviours. And what's unclear right now is how these different things weigh up on balance. How do we weigh, the negatives of a wild animal's life against the positives that it experiences? Because if we can't do that, we don't actually know what the line of wild animal welfare is. We can't even make this comparison. And so, thinking about how we weigh off these trade-offs, this is a nice figure that comes from Jake Vesey a few years ago, which was thinking about, the lines of welfare for wild and captive animals, and thinking about how we trade off perhaps these losses of behavioural freedoms and some of the psychological parts of well-being against, the increases in physical welfare that come from having an animal in captivity. And, perhaps the peak welfare is somewhere in the middle there. But obviously, if we're only setting the standard according to wild animal welfare, we wouldn't be able to sort of see this improvement. I also think that perhaps there's a little bit of counterintuitiveness if we do think about wild animal welfare. For instance, if it did turn out that, we went out and looked at wild animals and for the most part their welfare was quite low, do we still want that to be the threshold for captive animal welfare, that, look, their wild relatives do really badly, so it doesn't matter if we mistreat them in captivity? That doesn't really seem right to me. And also it's a little bit counterintuitive, I guess, that it'll give us different results for different species. It's almost certainly not the case that every animal out in the wild has the same level of welfare. They're going to be exposed to all kinds of different things. And again, if I was in a zoo and, someone came to me and said, well, look, it turns out that otters in the wild have quite high welfare because they have a lot of opportunities, they play together, they have fun, so we should invest a lot of resources in making their exhibit really good because we want to match their wild standing. But it turns out that elk actually have fairly poor welfare in the wild because, they have a lot of predators, they're very stressed, so we don't have to, put any resources into them, and our elk can just live in a cage out the back. I wouldn't be happy with that. I would want to say, well, look, we want to get all our animals up to sort of some standard of living that we think is acceptable, and we don't want that to be different. So perhaps a less arbitrary solution is thinking about the acceptable welfare from the point of view of the individual. So, in humans, we can think about, introspect or talk to others about what do we think is our level of acceptable welfare, when do we think intervention is included. I think for animals, it is a much trickier kind of prospect because we can't ask them. We can't say, well, what level of welfare would you be happier with? And so thinking through this, I thought about, are there ways to ask them? And this is very speculative, but we do have methods of asking animals what they want. And, there's a sort of, we have welfare measures that can tell us, welfare of animals. We don't necessarily know how they feel about their own welfare. This is kind of almost like a meta question to an animal. But perhaps there is some role here for preference and motivation testing. We are able to ask animals how much they value different resources. Perhaps there's some way of asking them how much they value welfare improvements implicita, not just related to a resource itself. And if we could see, say, animals work hard to improve their welfare up to a certain point, and then they don't work so hard after that, perhaps that would represent a line that's considered acceptable from the point of view of that animal, and that would give us a good reason to think about the fact that this might be what we would think is a threshold of acceptable welfare as well. And I've just noted here, obviously, this is a lot more difficult than it makes it sound if I say it in 2 minutes. We all know there are a lot of problems with preference and motivation tests in creating ones that ask the right questions, in interpreting them the right way, but perhaps we could make some headway doing something like this. So to finish up, I think there's no obvious privileged way of setting a line for acceptable welfare. There are times when we're going to want to say, does this animal have sort of good, poor welfare? Is it adequate? Is it acceptable? Do we need to do more? But there is a number of different ways of doing this, and there's not one that sort of stands out as the real best way. Each of these potential lines has different benefits and different drawbacks, and it's important for us to reflect on which one we choose and the reasons for its use. So I think that the main takeaway from what I want to say here is that if you are using levels of acceptable welfare, think about why you've chosen it and what the reasons for its use are, with reference to these benefits and drawbacks as well. And we could think about the possibility of using multiple thresholds too. So, thresholds for urgent intervention, below which we think welfare is just completely unacceptable, and perhaps higher levels that are aspirational, where we think welfare is good. And certification and labelling schemes certainly lend themselves to this sort of thing, that you can, have different categories of welfare conditions. But again, we need to be thinking about what those categories represent and where those lines sit. All right, thank you.
Thank you, Dr. Browning. Do we have questions in the audience? We have one right there. Are you able to make your way to the microphone there? And then we'll go there a second. Oh, actually, we'll go to the back first and then to you. Oh.
Hello. Hey, Heather. The asking the animal approach, is there a way for accounting for adaptive preferences with that? Because you could easily say, like, given the environment that the animal's now in, like their welfare, we can say that this is good or bad welfare for the animal, but that might not be sufficient. It might not enable us to determine, whether the condition itself is going to be optimal.
Yeah,
anyway.
no, I understand what you're saying there. I think one of the things with adaptive preferences may be that it applies to conditions that animals experience, perhaps not to the welfare level itself. So animals may adapt to thinking, well, I don't mind having a cage like this, I'd be scared being out in the bigger world. It may not mean that the animal prefers to have welfare at this baseline rather than the other one. That's something we'd probably need to investigate though. And I mean, one of the ways of doing that might be comparing animals from different housing systems and look at whether or not their baselines change based on that. So whether, animals in poorer housing conditions do actually have different preferences from other ones. But yeah, I think this comes under the thing where I said, there's all these, the well-known issues of preference and motivation testing, and I think that's going to be one of the things that needs to be accounted for too. Yeah, please.
Lovely. That's very loud. That was a really, really interesting talk. I was just wondering, how do you, when it comes to creating a general criteria for animal welfare, how are you going to take into account individual difference? Because obviously individual animals deal with the same circumstances incredibly differently, and how are you going to deal with that with social animals that are kept in a group?
Yeah, good. So if we've got this idea that animals have individual differences, I think those differences are going to be more at the level of what conditions are good or bad for their welfare than what their actual level of welfare is, if that makes sense. So, I mean, what that would tell us is that when we're thinking about what counts as the acceptable line of welfare for animals, we need to be using animal-based measures to check on that. So, I mean, obviously there's all these kind of resource-based measures that we often assess whether institutions are doing well or poorly based on whether they have the right inputs for the animal. But I think you're right that when you do have individual differences, some set of inputs might be at the acceptable level for one animal and not at the acceptable level for another. And this kind of emphasises the importance of using the animal-based measures then if we're going to be doing that. And perhaps if we can map out what the standard variation is likely to be in some particular population, we can start getting a sense of where that range of inputs needs to be.
Okay, thank you, Heather.
Thank you.
I'm pleased to introduce our next speaker, Dr. Sandra Baker from the University of Oxford. She's going to be talking about assessing animal welfare impacts. Take your time, I can talk slower. In wild Norway rat management. So whenever you're ready, thank you.
Right, good morning everyone, thank you for your patience. So I'm going to talk about an assessment of animal welfare impacts in the management of wild Norway rats. I've called my first slide a meeting of minds because this work was very much a collaboration. The first thing I did when I started the project was to gather together a group of expert stakeholders in a wide range of fields, including rodent management, rodent biology, animal welfare science, wildlife management, and veterinary science and medicine. And some of those people are here in this room today. And I think that the strength of this work lies very much in the level of agreement achieved among such a wide, diverse range of experts. And here is the citation for the paper that I'm going to talk about. I'll give you that again at the end if you're interested. It's published in Animal Welfare. So the main aim of this research was to assess the relative welfare impact of 6 different lethal rat management methods, so ways of killing rats. First of all, we looked at snap trapping, which is basically using the standard kind of Tom and Jerry style trap, which is intended to kill rats or mice, depending on which size you buy, by crushing the back of the skull or the upper cervical vertebrae. And it's crucial to recognise, in looking at the results of this assessment, that these traps are totally unregulated in the UK and almost everywhere else in the world. We also looked at the impacts of cage trapping, which is obviously a live capture method, but followed with a concussive blow to the head for humane dispatch. And also glue trapping, again, a supposedly live method of catching rats, followed by a concussive blow to the head. And we also looked at 3 different baiting methods, the first of all being anticoagulant rodenticides, which, as you probably know, act to disrupt blood clotting, and they cause internal hemorrhaging and external bleeding. We also looked at another poison called cholecalciferol, which is the naturally occurring version of vitamin D3, and it acts to increase intestinal absorption of calcium from the skeleton. So it mobilizes calcium from the bones and causes calcification of major arteries around the heart and kidneys in particular, and ultimately causes heart or kidney failure. And then finally we looked at non-toxic lethal cellulose baiting, which is it's provided as a non-toxic food source for rodents, and it interferes with the digestive system and ultimately causes lethal dehydration. So we had a couple of other aims as well. One was to identify ways of reducing the welfare impacts of these particular methods, and the other was to highlight any gaps in the data that we identified as we went along. So I haven't got much time to spend on the methods, unfortunately, although I think the methods are quite interesting. First of all, I drafted a set of standard operating procedures, one for each of the different methods. I did that by combining information from best practice guidance and from the wildlife and welfare legislation, and I also had some input from the pest controllers on my stakeholder panel. I conducted a literature review looking for any evidence for or against welfare impacts of the different methods, and ultimately facilitated a couple of online welfare assessment workshops where the stakeholders came together, and I sort of led them through the process of applying an animal welfare assessment model and encouraged discussion among them to try and reach consensus on a range of welfare impact scores, one for each of the methods, so they could be compared. Okay, so the model is basically a scoring system which uses data from the literature and expert opinion to help you to ascertain appropriate scores. Okay, so the model was devised by Australian government scientists Trudy Sharp and Glenn Saunders a few years ago, and they did that with expert stakeholders in Australia, and as a result of that, the model has sort of achieved a very high level of acceptability among pest controllers and wildlife managers in Australia. And the model's been used for all sorts of species in Australia, and it's also been used in New Zealand, and I've used it a couple of times before in the UK. So the model consists of two parts. Part A looks at the welfare impact of a method on an animal before any action that causes death, so basically any non-lethal components of a method. It's based on Meller and Reed's 5 domains model, and the premise of that model is that welfare impacts under each of 4 physical domains can then be used to cautiously infer welfare impacts under the 5th mental state domain. And that, of course, is the all-important one for assessing welfare. And scores under Part A are a number ranging from 1 to 8, with 8 being the highest level of impact. The other part of the model looks at the welfare impact of any killing method involved, and that's based on Donald Broom's criteria for assessing the welfare impact of killing methods, which he said was basically the product of the severity of any poor welfare before death or irreversible unconsciousness, and the duration of that poor welfare. So, yes, that Part B produces a letter score as opposed to a number, and the letters range from A to H. And the different parts of the model that are used will depend on the method being assessed. So if a method consists of lethal and non-lethal components, you'll use both. And both parts of the model use welfare impact scales, which are provided as part of the model, along with examples to help allocate categories for welfare impact and the duration of that impact. I also asked stakeholders to try and apply a confidence score to the impacts and the durations that they settled on, ranging from 0 to 3, with 3 being the highest level of confidence. So this is the format of the results. On the y-axis, you have the Part A results, which are numbers, and on the x-axis along the bottom, you have the Part B results, which are shown as letters. And as you move from the bottom left up to the top right, the welfare impact scores get worse. You can see that there are 4 grey shaded areas there. 1, 2 and 3 each relate to 3 different methods, and the area marked 4 actually is the score achieved by all 3 of the baiting methods. They all scored exactly the same, but for very different reasons. Okay, so I'm going to start with the methods that scored worse, so they're on the peripheries of the graph. First of all, looking at glue trapping, and glue trapping obviously had 2 components, the trapping and then the killing, but because the period during which the animals were being trapped, live trapped, had the biggest impact on their welfare score outcome. I'll talk about that because we're so limited for time, rather than talking about the concussive blow. So the stakeholders allocated an extreme impact, which was the worst category of impact, lasting for hours, and they had a high level of confidence in both those things. So the primary kinds of impact were health-related, domain 3, and behavioural, and so rodents are known to experience all sorts of horrible things during glue trapping. They basically get stuck to the glue, and the more they struggle, they get drawn and drawn into the glue, and they start to tear their skin potentially, break their own bones, chew through their own limbs. Their eyes and mouths can get glued shut, and they get covered in their own feces and urine, and they can die of suffocation or exhaustion, even though this is supposed to be a live capture method. Behaviourally, obviously, they're unable to do the majority of things they would normally do, and they're also potentially subject to cannibalism by the rats that are caught nearby on the same glue trap. And ultimately, the evidence suggests that they're likely to be experiencing anxiety, pain, fear, panic, distress, hunger, thirst, and breathlessness. So, and I'm going to talk about the 3 baiting methods, which, as I said, all scored the same. So the anticoagulants first, they scored a severe to extreme impact lasting for days. The stakeholders had a medium level and a high level of confidence in those 2 things. And the welfare impacts were largely health related, including hemorrhaging into organs and body cavities, reduced respiratory function if bleeding happens into the lungs, and death potentially by anemia or hypovolemic shock. And the animals are likely to experience severe pain, breathlessness, lethargy, weakness, thirst, dizziness, anxiety, and fear. And the evidence suggests that rodents remain conscious throughout that period of days while all this is happening. So next, cholecalciferol, which, as I said, scored the same. This time, the stakeholders had a high level of confidence in both those things, and the range of physical impacts was wider, including nutritional ones, health-related, and behavioural. And the health-related impacts included osteomalacia, which is the softening of bones, calcification of blood vessels around the heart, and that should actually say kidneys, and of course, behavioural restrictions. As they're affected by the poison, they're unable to forage normally or act to escape predators. And the mental impacts of this are likely to be pain, nausea, lethargy, weakness, listlessness, thirst, breathlessness, fear, anxiety, confusion. And again, the evidence suggests that the rodents stay conscious throughout. And then the third baiting method, cellulose, again, the same score, and again, the stakeholders had a high level of confidence in this. The animals are likely to suffer starvation and dehydration because the ingestion of cellulose interferes with their eating and drinking. They tend to experience quite severe gut obstruction as the cellulose accumulates in their gut and starts to draw fluid from the blood supply, and it swells further, and they're likely to experience hypovolemia and reduced blood pressure, tissue ischemia, which is reduction of oxygen to the tissues, and multi-organ failure and circulatory shock. And ultimately, they're likely to be experiencing gastrointestinal pain and discomfort, nausea, weakness, dizziness, hunger, anxiety, and fear. And again, they're thought to remain conscious. So, cage trapping. Again, as with the glue trapping, I'm going to focus just on the component where they're in the cage trap because that had the largest impact on their welfare. stakeholders allocated a moderate to severe impact lasting for hours, and they had a medium and high level of confidence in that. Impacts were largely behavioural because their movement is restricted and they're unable to do the things they would normally do, and they're likely to be experiencing fear and distress. Then finally, snap trapping. So stakeholders allocated the widest possible range of impact scores for this, ranging from a no impact all the way through to extreme, and they felt that that would be lasting for seconds to minutes with the animals still conscious. And they had a low and medium level of confidence in this. And the health-related issues, there's an awful lot of uncertainty about these with snap traps. Because they've been unregulated, a huge variety of shapes and sizes and strengths and designs have been able to proliferate, and there's uncertainty about the variation, therefore, in trap strike location and power, and therefore the injury type and the way that animals die. But possible candidate ways include hemorrhaging, paralysis, asphyxiation, occlusion of blood flow to the brain, compression of the heart and lungs, and hemorrhagic shock and heart failure. It's not known at what point, really, the animals are likely to become unconscious in any of that. Of course, behaviourally, they're stuck in the trap and unable to behave normally, and they're likely to experience pain, fear, and severe distress. So, moving on to the results now, the greatest welfare impacts, according to the study, were from the baiting methods and from glue trapping. And if you're looking from a purely welfare perspective, then you would say that these should be considered last resorts. Lower impacts were attached to cage trapping, and highly variable impacts associated with snap trapping, but a good snap trap could potentially be the most welfare-friendly of the options that we assessed. And we decided to write in the paper that given our findings on the dependency of the rodent control industry on anticoagulants, rat management may be one of the greatest anthropogenic impacts on wild animal welfare. So how can we use these scores usefully? Well, they can be used quite readily with existing frameworks in wildlife management, or in rodent management at least, to aid the consideration of welfare in rat management alongside other important considerations, including efficacy, safety, cost, non-target welfare, and public acceptability, and so on. The first of those is the Campaign for Responsible Rodenticide Use Risk Hierarchy, which is a set of stepwise measures for preventing and removing rodent populations, and that focuses on proofing and removal of harbourage, and also, when lethal management is considered necessary, using the least severe method that's considered effective. And then the Dubois 7 Principles for Ethical Wildlife Management, again, a set of stepwise measures or considerations which begin with changing human behaviour where you can to either reduce or eliminate or prevent the problem that's perceived, and further down the list of considerations to use the method with the least impact that's likely to work. I know I'm running out of time now, so I'll just race through this. So we identified some ways of potentially reducing welfare impacts in these methods. First of all, preventing or minimise population establishment is the obvious way. Choosing good quality snap traps. We're using cage traps with effective concussive blow to the head, where possible, over baiting methods or glue traps. Where cage traps are used, covering them or supplying them with food, water or bedding, and using any traps, checking them frequently or monitoring them using remote sensing. And a snap trap situation needs to change. They either need to be regulated, or a voluntary certification scheme would be extremely helpful, and there's actually one of those coming into force soon. Improved training in applying a concussive blow to head would also be a positive step. It's also been suggested in the past that it might be possible to develop rodenticides with analgesics, sedatives, or general anaesthetics. So I know I'm out of time. Is it right to just save my last few bits? So the data gaps we identified were that we need to know more about the time to unconsciousness in snap traps, and if the voluntary certification scheme takes off, then more data will accumulate, and I think that will have quite a rapid impact on the types of traps that are made and sold and bought. We need to know more about the prevalence of different welfare impacts experienced by rats in both snap traps and cage traps, and the impacts experienced by rats that are being transferred from a cage trap into a sack in order to undergo a concussive blow to the head. Some other potential improvements might include improved welfare training for pest control technicians, better informing the public, and better enforcement of food safety and health and safety legislation. And I'll finish by thanking my sponsors, one of which was UFAW, of course, another was the Animal Welfare Foundation, and also the Eleanor Patterson Baker Foundation. And the paper is available, as I said, in Animal Welfare, and it's available open access, so anybody can read that if they're interested. Thank you.
Thank you. We have time for one question if somebody wants to make their way to the microphone or raise their hand.
I'll throw one at them. What about...
you press the yellow button first.
Good point. What about releasing trapped rodents somewhere else rather than killing them in the cage?
So something that people often want to do, and they think will obviously be better than killing rats, is to use some form of non-lethal control. And I understand from talking to Hugh that members of the public often contact UFAW, and I know they also contact the RSPCA asking about live capture and release. And we decided not to include it in this study because there just isn't enough information on which to base proper assessments. But that's certainly an area that needs to be looked into, because you can't assume that just because you're not killing something that you're not impacting its welfare, obviously, or the welfare of other animals that are already in the place where you release the animals that you've captured. So I think that's an area that definitely needs more work.
Yeah, I think we can take one more question. If you don't mind.
Hi, Georgia Mason, University of Guelph. The appalling techniques you said should be methods of last resort and commonly used as methods of first resort. And I wonder what you could say about likely changes in policy or even the law that might get rid of these methods.
Well, this work was obviously done in conjunction with rodent controllers. And in talking to them throughout the process, it was very useful, I think, for us to hear their opinions about why they need these methods. And I think they are quite worried about this kind of work as well. In terms of policy, I think it's unlikely that anything will change very soon. And I think it always comes back to what are the alternatives. So it's a matter of finding something that's better. And I think the work with the snap traps is really important in that respect. Of course, I've heard from pest controllers that snap traps obviously aren't useful in all situations. There are some populations that don't go into snap traps. There are some places where you can't put them, and they prefer to put a glue trap because they can just slide it under a unit in a food processing plant or something like that. So those are the kinds of arguments that you get against change. But this gives me an opportunity to talk about the voluntary certification scheme, which has been spearheaded by the German Environment Agency. It's a piece of work that I was involved with over the last 2.5 years or so, and it's called No-KERO, which stands for No Chemical Rodenticides, and it actually stems from an effort to position snap traps as a viable alternative to rodenticides, which they actually transgress various of the criteria that should be used for a pest control method to be used in the EU, but on the grounds that there's no alternative, no viable alternative, they're sort of waved through. So the idea behind the No-KERO working group was that if traps could be positioned as a measurable alternative by producing guidelines that can measure their welfare effects and their efficacy, it would put them on a standing with being able to measure the welfare effects and the efficacy of rodenticides, and therefore it would become impossible not to consider them as alternatives. So the European Commission have been looking at the guidance that we as a working group produced, and they've decided that they're happy with it and that it does provide a suitable alternative method of examining another approach. And the German Environment Agency are already testing traps. So there are some snap traps that cause irreversible unconsciousness in rats within a few seconds now. A couple are mentioned in the paper, but I know that they're testing traps quite quickly now, I think. So hopefully that's something that will change very soon.
Thank you. Right, I will welcome the last speaker of the session, Giovanna Massei, whose the title of your talk is Fertility Control to Mitigate Human-Wildlife Conflicts and Promote Coexistence. So the floor is yours.
Thank you. Good morning. Thank you. I know I'm the last one of the morning, but just to wake everybody up. My name is Giovanna Massei, and I am the Europe Director of the Bas Rodenburg Institute for Wildlife Fertility Control. I'm also a researcher based both at the University of York and at the Animal and Plant Health Agency, and today I'm here to talk about the research in this field. I've worked for about 20 years in this field, and I'm delighted to be here. I'm delighted that the previous speakers touched on some key issues that I'll be developing. So you might be all aware that human-wildlife conflicts are increasing. They're increasing worldwide. This is a non-exhaustive list. And we're talking about conflicts in terms of environmental or economic damage. And that could be millions, billions, in fact, per year. And please keep this list in mind, because I'll be going back to this list in a second. So why the human-wildlife conflicts are increasing? Well, the first issue we have is people. There are too many people around the world, and if you look at the world human population, Europe, Southeast Asia are very, very highly populated. In addition, very recently, the number of people living in cities has surpassed the number of people living in rural areas. And this has very important implications. People living in cities have a very romantic idea on how to manage wildlife. We've just heard from the previous speakers, one of the most asked options, if you have a rat in your house or any fluid animals, is can we move it? Can we move it somewhere else? Because that's generally a quick fix, and that's what people would like to see. Because moving an animal doesn't mean killing it. The chicken is there because New York Zoo put chicken in their zoo just to show children how a Kentucky Fried Chicken looked originally. These people, so people living in towns have, as I said, very, very little exposure to wildlife, to free-living wildlife, but very strong ideas on how to manage wildlife. At the same time, we have created shelter for animals. We feed animals, either intentionally or not intentionally. We give them crops. Well, we don't give them crops, but they feed them crops. The forests are increasing, certainly in the developed world, so in Europe and in the States, forests are increasing. And this is a very good example of a hyper-successful animal. I worked most of my life, I did my PhD on wild boar. They're intelligent, they are fascinating to study. This on the y-axis is the number of wild boar killed or culled by hunters in 17 European member states in the last 30 years. And the take-home message here is that the numbers are growing. Until 2012, some countries were killing 500,700,000 wild boar per year. In 2019, Germany alone killed in excess of 800,000 wild boar in a year. Now, I'm quoting the wild boar because the wild boar, if you think about the list of human-wildlife conflicts, you can tick it all for the wild boar. The wild boar is a prime example of this. At the same time, the major source of mortality for wild boar, the hunters, are declining. The hunters are becoming an endangered species, and they're going down. So the difference between these trends in hunters and wild boar will mean that we will have a lot more problems. In fact, if anybody wants to work on wild boar in the future, there's definitely a future there in terms of jobs. Something else in terms of squirrels. So in the UK, again, you might be aware that the red squirrel is the native one. The grey squirrel is non-native. There are now 10 times more grey squirrels than red. The impact on red squirrels has been major, but also the impact of forestry. We're talking about 37 million euros per year by the grey squirrels on the impact. And this is a report from colleagues in forest research on attitudes, public attitudes to grey squirrel control. There are different methods. Blue and red means either very acceptable or acceptable. And contraception by almost 4,000 people was considered the most acceptable method. Possibly because a number of people thinking about contraception think about we will put squirrels or wild boar or anything else on the pill. Easy. Not quite. But a number of people also really disliked killing in any form. So if we think about wildlife, we have wildlife, I mean problem animals having an impact on economic or environmental impact. We have two ways of managing these populations, either lethal methods or non-lethal methods. We have actually published a paper several years ago on translocation of problem animals, not of endangered animals. If anybody wants a copy, I'd be delighted to talk about it forever. But in terms of today, I'll just focus on fertility control and show you some examples. This is an ideal list of what an ideal contraceptive would look like. And you can read it, but effectively like no side effects apart from the effects of reproduction, feasible, low cost, species-specific, no effect on the food chain, and can be delivered to a large proportion of the population. There are several contraceptives now available around the world, and I have mainly worked with GnRH vaccine. These are vaccines that induce they're injectable, and they induce antibodies against proteins or hormones that are essential for reproduction. The good news is that after 1 or 2 injections, the majority of animals become infertile for a number of years. So we do have some practical applications credible, but we're also trying to work out, and other groups in the world are working on oral contraceptives for obvious reasons. So some examples of our work. This is the first one in early 2000. Some colleagues from the States said they had this fantastic contraceptive, injectable contraceptive, or wild boar, would we like to try it? And we said, absolutely yes. However, we really want to know also whether there are any side effects whatsoever before going around and injecting wild boar in the wild. So we tested it. We had 12 animals treated with a single injection and 12 controls. And the summary of this is that 11 out of the 12 females injected with one single dose never reproduced, did not reproduce for at least 4 to 6 years, after which we stopped the trial, because it's very expensive to keep indoors in an animal hotel, in fact, 24 wild boar. But we could not find any side effects on the welfare, on the physiology, on the behavior. We tried every possible measure, and there were no effects. So we were very happy. We went to the field, and we injected a number of animals. The majority ended up shot dead by hunters. So it was a bit difficult to collect the data we wanted. But what we could find was that some animals had been treated when pregnant, and in these animals, pregnancy went to term, and then animals became infertile, which seems to be the case for other species. The other important part of the study is that because it generated interest, and the wild boar are in every continent, wild boar, wild pigs, apart from Antarctica, this generated 2 more studies, one in Barcelona, near Barcelona, that has just finished, and another one in Hong Kong. When we are doing this work, we were wondering whether, okay, if these are injectable, so expensive to deliver because you have to trap an animal injected. However, if we developed an oral contraceptive, how would we give it to the wild boar and to the wild boar only, given that this oral contraceptive is likely to affect any mammal that will feed on it? So we came up and we developed the BOSS, the Boar-Operated System, and I think it's quick. We were very worried. This was tested in the UK, and we were very worried about the badgers. We didn't want the badgers to feed on contraceptives. At this point, we did not have contraceptive. We just had plain bait to see whether animals were happy to use these. We tested it in captivity, and then we went to the field in England. So they're more than happy to feed from it. This is just maize. Wild boar are not yellow on their nose, generally. So the other one is coming, she's yellow too. We put a marker there, so we know that the animal has already been visiting this BOSS. And we now know, so we've tested this in several countries around the world, and we know that it works very well. The only animals for which it doesn't work, so the only animal that can access the bait is the bear in the States. So we said, okay, we'll call it Bear-Operated System, and that will work too. Other examples. So this is from my colleague Dave Cowan, feral goats in a national park in Wales. They vandalize gardens, damage plant communities that are in poor health, but they also have an iconic status. People want to see them. And there are limited opportunities to translocate or to adopt out a lot of goats. So they went in 1 or 2 doses of injectable contraceptive, reduced female reproduction by 83 to 92% for 4 years. So a very good story. Very similar, my work in Hong Kong with cattle. Hong Kong used to have 1,300 cattle free roaming. And again, because traffic accidents, translocated animals return to the place where they were translocated from. There's strong public opposition to culling, and surgical sterilization was difficult. So we injected these animals. We did a captive trial and then a field trial. We injected animals with 2 doses of gonocon, and we found that this caused infertility for at least 3 years. This is an example from New Zealand. This is surgical sterilization, but this is just to prove that on the y-axis, you have the proportion of infected animals, animals infected, possums infected with leptospira or leptospirosis. And if you sterilize males or females, the number infected decreases because you have less frequent mating contacts. So you do have a reduction of the disease transmission without having to reduce the population size. Fertility control is widely used in South Africa to increase the time between births of elephants. So all females contribute to fertility control, to population by breeding, but their intercalving period is extended. And this is, I think, the last project. This is a very big project that I'm leading in New York, 1 million project on developing and delivering oral contraceptives for gray squirrels. One of the interesting parts is that if you have 3 million squirrels, which is as important as delivering an oral contraceptive, how do you make sure that you can deliver to enough animals in the population to make a difference? So we use rhodamine B. Rhodamine B is a bait marker. if you eat it yourself, your hair under UV light will become this color, bright pink. So we made a lot of squirrels bright pink. And in this way, we have been able to demonstrate that the majority of animals in a population can feed on baits, that in the future will have the oral contraceptive. So when people ask me, when should we use no lethal control? The answer is when lethal control is any of these or a combination of these. And finally, we have some contraceptives available, not only immunocontraceptive, but also some oral contraceptive already available. We have modeled the impact of fertility control on population dynamics, but we are still developing more contraceptive. We are still looking at welfare and field trials. We are looking at the cost of sustainability, and we are looking at the legal aspects of using contraceptives. So in conclusions, fertility control will play a crucial role in mitigating human-wildlife conflicts. We, at the BOTS-TIBER, are there to identify species and contexts for which fertility control would make sense. And we need to manage public expectations because the public love it, but it's something that we have to manage. And I wish to acknowledge a number of people, including the organizers of the conference. Thank you.
Thank you. We have time for a question.
If you could go to the microphone. Press the button.
Hi, my name is Julia, and then I'm studying at the University of British Columbia. Hello, thank you very much for your presentation. It's really interesting. I just want to ask about, you mentioned about the boar hunters, and then you mentioned some of them actually killed some of the sample that you had. I just wondered, how did you communicate with them or approach them regarding the sample and whether a consensus was made? Thank you.
Yes, we are in fact working on talking to stakeholders before starting any work on it, but in this case, we couldn't reach all the hunters. Some of the animals had radio tracking devices, and we tracked the collar to the garage of the hunter. And they denied that they had the wild boar. I was like, no, actually, we know because it's beeping. So, yes.
Do we have a last question? I don't see any. Oh, yes.
Right at the top. Can you...
Hey, so I was wondering how much work is done, and if not a lot, then are you doing work on, I guess, negative effects of contraception, like delivered to the young? So, you know, in that video, the young were accessing the food as well. So I'm wondering about, delivering it to young, and then also, we know that at least in humans, contraception can be really damaging, like mentally and physiologically and stuff. So I was wondering how much like work there is about that, or like considerations of those kind of effects that you might not see.
Yes, thank you. The effect on the young, some of my colleagues in the animal welfare unit were very concerned about the weight of it falling on the heads of the piglets. The piglets couldn't care less, so that was fine. But certainly whenever you develop a contraceptive, you would test it, or I would recommend to test it in captivity on different age classes. So that's the first thing. In terms of welfare, there is not a lot done. We should be doing more. We do try to do everything we can with captive studies to start with when we start with a new species. But we should be doing more, but then we should put it in the wider context of how do we compare this with shooting or with translocating or with any other method of reducing human-wildlife conflicts. And that's the meaningful comparison. Thank you.
So that concludes the first session. Thank you to all of our speakers and to you for your attention. And I believe we have a break until 11:30, and then we meet back here. So thank you.
Steve? No sign of Katie still? No, just... Yeah, go. Oh, where's Peter gone? Oh. Where is she? No, you can sit next to Louisa, but Peter was over that side. Okay, thank you everyone. Thanks for coming back. I hope you're suitably caffeinated. It's great to see you all back in here. So without further ado, I'd like to introduce our next keynote speaker, continuing the theme of tackling really small issues in animal welfare, or actually really enormous ones. I'd like to introduce Bas Rodenburg from Utrecht University, who's going to be talking about meeting the demands of a growing human population while also achieving good welfare. So best of luck solving that one in 40 minutes, Bas.
Thank you very much, Hugh, and good morning, everyone. Indeed, thanks for the organizers for providing me with this very modest title. So I hope to address these topics in the talk. As we already saw this morning in one of the other talks, we all know that the world population is growing. Since the 1950s, a steep increase in the number of people in the world. And of course, this leads to an increase in food demand for humans, and this results in a demand of animal-based food and plant-based food. At the same time, this food production is also an issue for the planet and for the sustainability of the planet, where you see that food production is responsible for about 25% of greenhouse gas emissions. And if we zoom in on consumption of animal-based food, of course, probably we all know that large differences exist in meat consumption across the world. And this, I think this view of the world from our World in Data on global meat demand tells a very powerful message. It shows that how much meat should be produced if we would eat like the average citizen of a given country. And at the moment, you see by the yellow arrow here at the bottom, you can see the current meat production in the world. And if we would go to the dark red level in the US, the Americas, Australia, and also the bright orange in Europe, we would need twice as much meat produced as we do at present. And of course, this is very unlikely to be feasible and will not be sustainable. Also, if you look at this map in a development from the 1950s, there's been a sharp increase in meat consumption. And this also shows that compared to the 1980s, there's a huge increase in meat consumption. And at that time, I think people were also able to feed themselves, to be healthy, and to be thriving. So probably we're eating also more meat in some parts of the world than is good for us. And this is not sustainable, especially also this Western style diet is also associated with increased health problems. And of course, we have the animal side. With all these animals that are kept for meat production, there's a risk of poor animal welfare due to intensive production. So I think this shows that we need to rethink our intake of animal-based food. So if we look at the question that I was asked to address, how can we meet the demands of a growing world population while also achieving good animal welfare, or more broadly, how can we do that in a sustainable manner? I think all the evidence is pointing in the direction that a transition is needed towards a system where you are producing food in a more circular way, where you're less dependent on external inputs, such as fertilizer, concentrates, pesticides, and where you also think about reducing animal production in areas where it's now very intensive and very much dependent on external inputs, and preventing nitrogen pollution. The map here on the right is showing the world map with countries over-applying nitrogen without associated gains in crop yield. And there in the red countries, and it's probably not visible, but there's a tiny speck in the middle that is the Netherlands. That's also bright orange. Of course, we like orange, but in this case, we could do with a bit less orange. So that's all indicators that change is needed. So if we think about these circular food systems, an important group working on this topic has been the group of Imke de Boer in Wageningen. And they've asked the question, how can we make our food system more sustainable, still supply sufficient food to feed the humans in the world, but also rethink this question of animal-based food. So probably there should be a shift, an increased intake of plant-based food by humans, and basically all the crops that are produced and that are edible by humans should also be used for human food production. And the animals should only be fed the leftovers that cannot be eaten by humans or grazed on marginal grassland. That's depicted in this figure here. And that will lead to a smaller size of animal production and animals more as recyclers in the system, rather than animals that we feed our grain or different grains that are growing in a crop area where you could also harvest crops for humans. They also looked at the most sustainable system from a protein perspective, to look at the amount of animal protein and plant protein. Humans need about 60 grams of protein per day, And they came, it's in these, you can see it in these graphs in the middle. They looked at how many grams of animal protein lead to the lowest land use for animal production and the highest availability of arable land for plant-based foods. And this is about 20 grams per day. So a diet with 20 grams of animal-based protein was found to be the most sustainable. What about animal welfare? Because it's not a given that animal welfare would improve by this transition to circular agriculture. There are concerns about feed quality. Feed quality may be more variable. Different feed sources may be used and they may be different throughout the year. There are risks to circularity, where, for instance, toxins might accumulate, microplastics can accumulate, all kinds of concerns on that side. And also, this whole transition is more focused on environmental sustainability, and the animal perspective is so far not really taken into account. The animals' interests are not central. So I think improving animal welfare should be an integral part of sustainable animal production, and I think, this is also a view that is supported by an increasing group of scientists. And to do that, I think we need to take a one welfare approach, where we recognize that animal welfare, human well-being, and environmental health are interconnected. And this topic is recently receiving more and more attention. Recently there has been an open letter with regard to the Stockholm Conference on Sustainability, where experts have called on governments to include animal welfare in sustainable development governance. But because so far animal welfare was not a goal in itself, and in that sense it can easily be forgotten in the whole sustainability discussion. It's also illustrated by this study from Linda Keeling and colleagues, where they studied the links between animal welfare and the different sustainability goals, and they found that it was linked to two of the sustainability goals, 12 and 14, focusing on responsible consumption and production and life below water. But also from the titles of the goals, it is clear that it's more in an indirect way and not really focused on the animal's interests. We can see everywhere, I think, that animal welfare is becoming more important and is receiving more attention. In the Netherlands, we recently have a report from the Council on Animal Affairs on humane livestock farming. This report was requested by the Dutch Ministry of Agriculture, and they were asking the Council to describe what would humane livestock farming look like, and how could positive animal welfare be included. So instead of focusing only on welfare problems, how can we also include the positive side? And the Council formulated 6 principles for humane livestock farming, adapted from the 5 domains model. So here's an overview of those six principles. It starts at the basis with respect for the intrinsic value of the animal and recognition of animals as sentient beings. Then, of course, the domains related to survival and to good feeding, good housing, and good health. In this model, the natural behavior was given a more central position, because it's also recognized that to adapt to an environment, animals use their behavior to come to a state that they regard as positive. So behavior is really central in their adaptation, in finding food if they are hungry, in finding shelter if they are not happy with the climatic conditions, in addressing health issues that they experience. And in the end, this leads, if the animal is successful, to a positive emotional state. And it also fits to the welfare definition that we use in Utrecht from Ol and van der Staaij, where an animal is able to adapt to the circumstances in its environment to reach a state that the animal perceives as positive. So that fits well together. This report on humane livestock farming was adopted by the Dutch government, and the government plans to take the lead in developing an agreement with stakeholders to improve animal welfare in livestock farming. And these 6 principles should provide the basis to look at systems and to look at developments needed in systems. It's more about also meeting the behavioral needs of farm animals, adapting the system to the animals instead of the animals to the system, and also on banning mutilations such as tail docking in pigs and beak trimming in laying hens. The beak trimming is already prohibited in the Netherlands. But we also should think back to this introduction on environmental sustainability. We should take an integrated approach. This map is to show you that the Netherlands does have some mountains. Maybe you thought the Netherlands were flat. But below this, the top graph is the number of cows per square kilometer in Western Europe, and the bottom one, the number of pigs. And below those 3 mountains is the Netherlands somewhere. So this is a real challenge, and also at the EU level, the country is asked to do something about this. So the current focus in the Netherlands is very much on reducing nitrogen pollution, and approximately a 30% reduction in the number of animals is now foreseen. And this transition, I think, also provides an opportunity to think about animal welfare and to redesign systems in such a way, where we also follow this one welfare approach, and we move towards systems where we have good animal welfare, good human well-being, and reduced environmental impact. And I think there's increasing evidence that also improved animal welfare and animal resilience can contribute to sustainability, and can contribute to this fact that animals can adapt to their system. That a stimulating and varied environment, the system that we're moving towards, will enable the animal to use its capacity to respond to challenges. This is an example from a study by Ingrid van Dixhoorn and colleagues from Wageningen, where they had enriched house pigs and barren house pigs, and they studied, for instance, damaging behavior, but also health after slaughter, looking at the lung lesions, and they saw that the enriched pigs showed less stress-related behavior indicated by biting and mounting, but they also had, after a sickness challenge with a PRRS infection, that's shown in the graph here. So on the right-hand side, you see the control animals, so there's no lesions there. On the left-hand side, you see the infected animals, and the barren housing clearly has higher lesion scores than the enriched housing. And so this also shows that this enriched housing is enabling the animals to cope with some of the challenges that they meet. And to sum up this first part, I think we see that a transition is needed towards a more plant-based diet in order to meet the demand of a growing world population. Animals can valorize leftover streams in the food chain and graze on marginal land. I think transition is a chance for improving animal welfare at the same time, and providing animals with a more stimulating environment contributes to sustainability. And I want to focus a bit more on that last point in the next part of the presentation. And that's describing a project that is recently finished at Wageningen, focusing on resilience in pigs. It's called Smart Resilience. I will tell a little bit about the main results from that project. And it's led by Dr. Liesbeth Bolhuis from Wageningen. And resilience we defined as the ability of an animal to recover from challenges. And we see that poor resilience increases the risk of welfare problems, such as lameness, respiration problems, tail biting, and aggression. And improved resilience should result in improved welfare, reduced antimicrobial use, and a reduced need for mutilation, such as tail docking. So in this the experiment that I talk about today, we had 3 batches of pigs. There were 96 pigs. The tempo times topics 20 cross, 6 pigs per pen, half for male, half for female. We used 6 behavior tests to look at the pigs' personality and behavioral characteristics, and we subjected them to 5 resilience challenges that were aiming to be similar to challenges the pigs would encounter in practice, so things like transport stress, heat stress, mixing, etc. We had two systems, so 48 pigs were in this alternative housing system and 48 in a conventional housing system. I'll explain these two systems. So the difference between housing systems started already at birth. So half of the pigs were, it's a chicken person talking about pigs. So half of the pigs were born in this conventional farrowing crate and weaned at 4 weeks of age, and half of the pigs were born in this family system. So they also started in a farrowing pen, but after a few days they were released and the piglets could mingle and the sows could mingle, and they lived in a group of 5 sows with their piglets together for 9 weeks. So weaning was also later. And at 9 weeks all the pigs were moved to Wageningen, and the treatment was continued. So the conventional pigs stayed in a conventional pen here on the left, with only the legally required enrichments. And the enriched pigs moved to a much larger pen with straw and wood shavings and peat moss and all these nice toys that were varied in time. And so it's sort of a pig paradise. Then here you have an overview of the time plan. So on the top you can see the alternative system with the pig staying in the family system for 9 weeks and then moving to Wageningen. Conventional system, farrowing crate, and then after 4 weeks a nursery pen, conventional. And then here at the bottom you can see the challenges from 12 to 21 weeks. So transport challenge, sickness challenge with LPS, heat stress, a small wound recovery, and in the end, slaughter and dissection. So if we look at the main results across this large experiment, we saw that the pigs from the alternative housing system had a quicker return to baseline after transport and after an LPS sickness challenge. If we looked at cortisol accumulation in their hairs, we also found an effect that's shown in the graph here on the right. So you see, this is the first 11 weeks, so that's basically the phase that they spent with the sow, so not in the experiment yet. There we saw no difference in cortisol in hairs between the treatments, but once they moved to Wageningen and were experiencing the different stressors, you can see that the conventionally housed pigs had higher cortisol levels in their hairs than pigs from the alternative system. They also had less wear and tear on the body, the pigs from the alternative system, and they had a better cognitive resilience, and this was tested by putting them in a problem-solving task after a stressor, after being socially isolated, and the alternative housed pigs were able to solve this problem easier after a stressor than the conventional pigs. In the same project, we are working on automatic monitoring of activity of the pigs. So here you can see an enriched pen and a barren pen. It's just a coincidence that the enriched is much more active in this video. So Lisette van der Zanden, for her PhD project, developed an algorithm to track the individual pigs. And this way we can also monitor individual activity levels. And we can also look at how they respond with their behavior, with their activity to a given stressor. This is an example of the response to the sickness challenge that's just being published as we speak, where Lisette looked at the recovery from this sickness challenge as a measure of stress resilience. So on the top you see 2 graphs of LPS injected pigs, and on the bottom 2 control pigs where you see no response in activity, and in these LPS injected pigs you see a dip in activity following the challenge. And if we compared the 2 different treatments, we saw that the enriched pigs had a slightly deeper dip, but also a faster recovery than the barren housed pig. So also in this challenge, they recovered faster, and this was also supported by the physiological measures that were collected. So this is a promising way to monitor animals continuously. When we look at pig farming in the Netherlands, there are an increasing number of farms also moving in directions like this. Things like increase in loose farrowing systems or only created during the first few days. environmental enrichment, providing jute sacks, some straw, a sand floor like the sows here in the picture, other materials, also things like practical welfare checks for farmers to be used by the farmer are increasingly popular. Antibiotics use is going down. There's increasing interest in keeping pigs with intact tails and a lot of support to move in this direction. And of course also castration has been phased out. So there's a lot of development going on, and this can be supported. Also in broilers, there's been a lot of changes in recent years in the Netherlands. If we look at the fast-growing broilers, they were not really accepted anymore by Dutch society. And there's been this shift for the fresh broiler meat market to move to slower growing genotypes. And recently, this study by Ingrid de Jong and colleagues was published, comparing a large number of flocks from this, the conventional fast-growing broiler with this intermediate system, the Dutch retail broiler, which has a growing period of 49 days and a slightly lower stocking density and some enrichment. And the Animal Protection Organization's Better Life label, Better Life 1 star, which is grows for 56 days, so 8 weeks, and is also given a covered veranda as enrichment for foraging and dust bathing. And they looked at a large number of flocks, so almost 1,900 flocks per system, and they looked at animal-based and resource-based welfare indicators. So mortality, foot pad dermatitis, hockburn, breast irritation, scratches, and on the resource-based stocking density, early feeding, and environmental enrichment. And this figure shows the welfare scores for the 3 systems. The lightest is the Better Life scheme, and the one in between, the retail broiler, and the darkest is the conventional broiler. So if you look in the top left of the figure, we can see that the total welfare score was indeed highest and best for the Better Life label. Conventional flocks had the lower scores, except for scratches. But you can also see that there's a large variation within systems and also an overlap between systems, especially for the animal-based measures. If you go down in the figure, the environmental-based measures are excluded from the data. And if you look in the bottom right, you can see that the systems are really overlapping. So that also tells us that farm management and also tailed chick quality are very important for welfare of a given broiler flock. So we should be careful when we move to high welfare systems that this improvement in welfare is also realized. Of course, also these welfare measures were mainly focusing on welfare problems and not so much on the behavioral aspects, but it is an important message. And it reminded me of the study from Marion Dawkins' group, 2004, where they studied the relationship between mortality and stocking density in broilers, also in a large number of broiler flocks. 80% of the UK broiler industry participated, and producers housed broiler flocks at these different stocking densities. And only in one example, there was some increase in mortality with increasing stocking density, but mainly you see a huge variation between producers and even between barns. So this shows you that flock management and farmer experience are really important in making, keeping animals in a successful way. A final example in this vein is from laying hens, also a very large study which focused on the relationship between housing system and mortality. In this figure you see the percentage of cage-free systems in green and cage systems in red, and you can see in many areas of the world cage-free systems are picking up. But one of the criticisms on these systems is always that they lead to higher mortality than cage systems. And this study asked, is this really still true in current day circumstances? They looked at 6,000 flocks, 176 million hens in 16 countries, and they compared mortality in conventional cages, furnished cages, and cage-free systems. And they saw that with increasing farmer experience, mortality is actually decreasing, also in the cage-free systems, with 0.35 to 0.65% per year. And if you look at countries that already have a long tradition of cage-free systems, such as France and the Netherlands, I added some orange to make the flags are quite similar, so then you can see the difference. But the blue line are the cage-free systems, and you can see that over the years, mortality is really decreasing in these systems. So that it's probably not the housing system, but it's more the experience of the farmer to work with these systems. So if we look at this second part in the Smart Resilience project, we saw that providing a more stimulating housing environment supports resilience of animals and animal welfare. I showed an example that automatic monitoring of activity and behavior can be a powerful tool to obtain insight in resilience and welfare over time. Also, we see with transition towards new systems that farm management and farmer experience are key. So we should also really be involved in advising producers and sort of co-developing new systems to solve problems early on. A question for me that is remaining is also how can we stimulate the transition towards more welfare-friendly systems, also in this bigger picture of sustainability and also, for instance, the issues of nitrogen pollution. Because if I look at the farmer perspective, I see that there's a lot of will to change, as long as there is a viable business model. In the end, they also have to make a living. They're often already quite engaged in a multi-million farm that they have there. So any solution that has the likelihood of being taken up in practice, also needs to work economically. And what a lot of people in practice also give back is that they also really look for this integrated approach, that we don't start now solving the nitrogen crisis and then move to animal welfare, and then every 5 years they have a different aim and a different message. So we really should think about doing this integrated. And then you can think, of course, about ways how to allow farmers to keep, for instance, half the animals they keep now and still have a viable business model. Of course, it's up to us as consumers to maybe eat less animal-based products, but pay a higher price for them because you know that they are being treated well. Well, in this audience, I'm quite convinced that would work, but worldwide it might still be a challenge. So you can also think about subsidies to allow farmers to keep fewer animals in a welfare-friendly systems. You can think about rewards for realized animal welfare, for increased biodiversity on the farm. And there's a lot of smaller initiatives in this vein already, but probably this is also something that's needed, for instance, at the EU level to think about this. And in the end, we can come back to this, the One Welfare aims of improving animal welfare, human well-being, and environmental health altogether. Of course, also food labeling could be used to inform consumers, to allow them to make informed choices. Things like an animal welfare label, also this method of production plus, like the Better Life scheme in the Netherlands, where you can give an increasing number of stars for increasingly welfare-friendly systems. Because in the end, it's really us as consumers that have a very large say in the future of animal production. And also, for industry, it's important to have this level playing field in Europe, for instance. So also the farm to fork strategy, where we look at this at a European level, I think is very important to make it also honest to the current farmers to move in this direction. To conclude, can we meet the demands of a growing world population while also achieving good animal welfare? Yes, I think we can. But there is a significant transition ahead in European animal production, and probably also worldwide. But I think especially the areas that were red on my first map need to think about their course, because they have a large influence on this whole picture. It's important to continue the dialogue with all stakeholders. There's in the Netherlands, for instance, now a lot of protesting by farmers and a lot of extreme views being shouted at each other. I think we need to get back to the table and to discuss what is the future and how can we get there. And it's important to realize the central role of farm management and farmer experience and take farmers along in the transition. And with that, I would like to thank you very much for your attention.
Thank you, Bas, and thank you for ending on such a positive message and for setting us up nicely for a debate about animal welfare labelling tomorrow. I can see one question up there, if you could come across to a mic.
Thank you very much. My name is Ron. I'm from Weizmann Institute of Science in Israel. I have two questions in one. Regarding the pig heaven, you mentioned not about castration, so the piglets were castrated, and if yes, using sedation or anesthesia?
In our case, the pigs were not castrated.
They were not castrated. Okay. And then later on, you mentioned that in the Netherlands, you're phasing out castration. So what are the alternatives? Is immunocastration, castration with sedation, anesthesia, painkillers?
At the moment, the main direction regarding castration in the Netherlands is to slaughter pigs at a lower body weight. So before the problems become apparent.
I see.
So there's no castration and no alternative methods being applied.
I see. So they are not even human nose, any other measures for boar taint?
No, and I think, but I'm not an expert in this area, so I think also the methods for detecting boar taint at the slaughter line have also improved in recent years, because it's as far as I talk to people in the pig industry, it's not a big topic anymore, and I'm not sure why that is.
Really? But you has the 2018 agreement to stop castration or ban castration in the European Union. But to my understanding, there's no real alternative to, like for the online or at-line detection of boar taint.
Okay, yeah. So I cannot really answer that, but at least in the context that I have, they don't indicate this as a major issue. When it is about boars, it's mainly about negative behaviors, mounting, biting, aggression. Those are still bigger issues. But from the meat quality, I think by moving the slaughter dates a bit earlier, they seem to have tackled this.
Thank you very much.
Okay, any other questions? Yeah, I can see a couple up there, whichever one you can get to first.
If you could come to actually the microphones, it's better, because that also means the people who are watching online can actually see you.
And say who you are at the start as well, please.
Thank you very much for this interesting talk. Masumet Ajipur from INRAE. You talked about business model for farmers. So I have rather a general question about what is the ideal business model, because if we think about, let's say, labeling farms with welfare improvement, welfare plus farms, means that consumers should pay more, so there we will have another label beside organic, etc. So consumers should pay more, and we will leave this choice of having farmer welfare plus farmers against normal farms. So I think it's a real question how we can really motivate farmers to go to be able to have a label, but for all farms and not leave it as a choice. And just to finish, I think the association that you made at the beginning with resilience, welfare plus farms have more resilient animals. Maybe it is something that can motivate farmers to have more production performance if we respect welfare. So it's just a general question, and see if you have any idea about.
Yeah. So for the first part on the labeling, you see in some countries that there is quite a good welfare label or method of production plus label, like the Better Life label in the Netherlands. Also other examples, Freedom Foods in the UK, Denmark, Germany. There's different examples, and those are also increasingly well recognized by consumers. So people who care about animal welfare in the Netherlands, they all know about the Better Life label. And last week they published a study that when people are shopping for barbecue meat, 20% is looking for the Better Life label, and of course many others are looking for the price. So that's a discussion you have, but I think that's a slow transition. at some point you might also decide as a government that, okay, better life 1 star or similar becomes the basis, like with the cages for the eggs, for the laying hens, where you at some point see, well, all consumers are happy to pay for eggs from cage-free systems, so we don't sell eggs from cage systems in the supermarket anymore. So I think that's a way to make some progress. And at the European level, they're now also discussing either a European welfare label or, because the risk is that you have 500 labels and that nobody finds it anymore and recognizes it. So it should be quite simple and accessible. You could also think about a one welfare label where you take also the holistic view into account. Yeah. And the second part?
The association between resilience and welfare.
Yeah. Yes, it's good, because you often see also in these like with the pig experiment that they also have a very good performance. These pigs in the enriched pen. So that could also be an argument to also show that at least it's not hurting production. Yeah.
Sorry, you need to talk into a mic, else the people who are listening online can't hear the question.
Then the question is, are there enough evidences for association between resilience and welfare? You showed results on the project, but now in the literature we don't have enough results.
I think that's still building. It's now quite a hot topic in different farm animal species to look at animal resilience and also farm resilience, to think about the whole system. But it's still relatively early days, so it's building. Yeah.
One quick one, if someone could come to the microphone. There's one down at the front down there if you want to come down. Sorry, this one down here. Yeah.
Thank you very much. Well, it's very interesting to hear also about the Smart Resilience project. I tried to be quick. I'm Kristina Kull from POCU in Vienna. So my question is regarding the numbers that you also mentioned, 9 to 20 grams of animal protein per day, it would total to around 7 to 8 kilos of animal protein in a year. And if we look at the numbers, it's very different, right? 60 to 80 kilos and so on. And to add also to the previous question, how to motivate the farmers, I'm wondering how can we make it happen so that all the farms that would stay would be exactly as we saw, the resilient farms. So the ones where the animal welfare is actually there. And do we need any stronger initiatives, perhaps from governmental boards? We set a limit of, if you produce more than 10 kilograms per person, you pay a fine or something like that. So more drastic measures. I'm wondering about time and feeding everyone, not just some. If you have any thoughts about these suggestions.
Yeah, I agree. It's a huge challenge, and there's no easy, quick fix, unfortunately. And at some point, it might be necessary also for governments to take more drastic measures and to put more pressure on the system and maybe start punishing the polluters or I always believe more in positive change, but it might be too slow and it might take too much time. So I hope that the farm to fork strategy will deliver some of these impacts in Europe. But yeah, it's a big challenge, and I think all of us are needed to help with that. Yeah.
Okay, let's stop there just to try and stay on time. But thank you, Bas. You'll be around, won't you, to answer more questions? Okay, so now we're going to move to trying to cram even more science into your day by doing our little batch of short talks. So these guys are going to be doing little 4-minute talks on some studies that they've done, and several of them also have posters if you want to get more information if we can't get all the info into 4 minutes. So first up, I'd like to welcome Jasmine Clarkson from Glasgow, talking about gradual decompression as an alternative to carbon dioxide.
Thank you very much.
So unfortunately, a prerequisite to the use of laboratory animals is that they must be humanely killed upon completion of the scientific work, which in the UK is regulated by Schedule 1 of ASPR. And from those listed in Schedule 1, exposure to a rising concentration of carbon dioxide gas is one of the most commonly used for laboratory rodents. However, despite its widespread use, there are significant welfare concerns due to its ability to induce a number of negative sensations, such as aversion, anxiety, breathlessness, and even pain at higher concentrations. And therefore, in recent years, it's been highlighted that there's currently an urgent and unmet need to find a humane alternative to carbon dioxide for killing laboratory rodents. So our project is systematically exploring the potential of gradual decompression, and this induces hyperbaric hypoxia, which basically occurs at low atmospheric pressures, whereby as the atmospheric pressure falls, there's also a proportional decrease in the availability of oxygen. So essentially, it's the equivalent of ascending to a high altitude. And importantly, for our research, hyperbaric hypoxia is considered insidious in humans, and therefore this is why we're exploring its potential in this context. So as I'm sure you can appreciate, a lot of killing methods are not instantaneous, and therefore we must derive what happens to the animal before it loses consciousness in order to infer its likely welfare impacts. And therefore we need to be able to determine the time taken for the animal to lose consciousness, their potential to experience potentially negative sensations, and also whether these negative sensations can be ameliorated by things such as drug interventions. So the aim of the data I'm showing today was to determine the behavioural consequences of both decompression and CO2 and determine their likely welfare impacts through the application of pharmacological interventions. So to do this, we conducted a 3x3 factorial designed study exploring both treatment and intervention. So we directly compared decompression with CO2, and we also included a sham treatment where the mice were exposed only to the chamber itself to obviously control for the potentially stressful effects of being in the chamber separately from killing. And then within each treatment, we had analgesia and anxiolytic treatment interventions to help infer whether any behaviours that we observed were actually mediated through pain and/or anxiety. So just a really brief kind of key findings. There are more on the poster that I'm presenting, but we found that compared to gradual decompression, mice exposed to carbon dioxide gassed more, which was also mitigated through the provision of analgesia, and therefore could indicate negative sensations such as breathlessness. You'll also see that mice undergoing CO2 performed more escape attempts, and interestingly, these occurred in saline-treated mice. We did find more mice ear scratching whilst undergoing decompression, but this also occurred in sham mice and was also unaffected by anxiolytic or analgesia. In terms of the more typical species-specific behaviours, we found that despite longer induction times, mice exposed to decompression exhibited more of these typical behaviours than those undergoing CO2. So to conclude, gradual decompression did take longer to result in unconsciousness and death compared to CO2. However, it is associated with fewer negative behavioural indicators of pain or anxiety, and therefore could offer a possible refinement. Thank you very much.
We're going to do questions with all four speakers at the end of this session, and thank you for keeping the time perfectly. So now, next up it's Holly Vickery talking about farmers' views on changing management practices for goat kids reared away from their dams. Holly.
Okay, so today I'm going to be talking about one of the aspects of my PhD. And a little bit about the background as to why I'm working on this particular area. Essentially, we know that with goat kids, the milk feeding and weaning stages are the highest risk period, and the highest levels of morbidity and mortality occur within this stage. The research is focused on adults, and little is known about kid management. And actually, understanding farmers' views and the things that they can do on farm is essential to our research being used in practice. So what I did is I created a survey that had two parts. And the first part was all about rearing methods. Essentially, what are you doing now? Whereas the second part was actually about what could you change? Why can you change it? And why can't you change it? So it was anonymous, recall-based, and we distributed it in person and online in 3 languages to try and widen participation. We used Likert scales and open text responses, as well as some option-based questions. Then I did some thematic analysis of those open texts, and then did Friedman tests on those Likert scales. So what did I find? One of the things that we're concerned about is abrupt weaning, particularly from these ad libitum milk feeding systems. So these are systems in which the goat kids can access the milk at all times and drink as much milk as they want. And we know that in calves this is concerning because they have slower rumen development because they're not eating as much solid feed. But farmers were really consistently unwilling to use any of these practices. And when we asked them why, the thing that really came up was that actually we have machines that feed all of the kids, and all they do is mix powder and water, and it just supplies lines of kids in different pens. So we can't change anything on the machine level because it will affect all of the pens of kids, and therefore they have different ages and we can't wean them all at once. So that was their really big concern around changing to any sort of gradual weaning method, and it's something that we need to address if we want to propose methods of gradual weaning that they can actually use. Then we asked them about the willingness to supply enrichment. And actually what we found here is that there was a huge difference in this median, and they were really willing to give them things that the kids could climb on top of. That was fine. That was really acceptable to them. Whereas loose items within the pen, that was kind of okay. They were reasonably willing to do that. Swinging items were a big no for these farmers. They did not want to give the kids anything that was hanging or swinging. It was just something that they didn't want to do. And this is because of safety. And one of my farmers said, well, goats make a really determined effort to find inventive ways to kill themselves or injure themselves. And this was a theme that came up a lot, and it was my favourite quotes. We had things like, goats are an accident waiting to happen. Why let them kill themselves after all the bloody hard work? So this was the main concern around enrichment. Farmers were really, really worried about the kids injuring themselves. And it seems that if we want them to enrich this early life environment, which we know is important for a wide array of purposes, we need to be giving them things that we can say are safe for the kids to use. So in terms of discussion and conclusions, perceived negative impact on health is a real barrier to farmers changing. And this makes sense because we do know that this stage is where they have a lot of immune changes, it's got timings on, and immune developments, and therefore the morbidity and mortality is highest during this stage. Health is an issue. And if we want to make changes to gradual weaning, we need to be able to show that it is beneficial in terms of health. They were really concerned about them scouring and about them force feeding. So if we took the teats away, it's got timing, then they thought that when we put the teats back, they'd drink too much milk and they'd scour and bloat. Feasibility is crucial. It needs to be pen level. They've got this rudimentary technology with these machines that feed multiple pens of kids. And what I really found throughout this is that participatory engagement is essential if we want farmers to use our strategies. I needed to find out from the farmers what they could actually do in order to be able to tailor my research into strategies that will be able to be applied on farm. So thank you very much for listening. And if you want to read more, it's just been published as a paper. And I also have a poster, which is at stand T9. Thank you.
Thanks, Holly. So next up is Miriam Zemanova from Fribourg in Switzerland, talking about animal welfare and the 3 R's in wildlife research.
Thank you. Hello, everyone. So I might be an outlier at this conference because my background is actually in ecology and nature conservation. And the aim of this talk is to raise awareness about the fact that wildlife research might be detrimental to the welfare of animals that we are using in our studies. So just here are some examples of traditionally implemented methods in wildlife research. So when we want to collect DNA, we might take blood or tissue samples. To estimate population sizes, we would mark the animals, for instance, through toe clipping. And for species detection, animals would be captured sometimes through lethal means. So these are effective methods, but the problem with them is that they have negative impact on animal welfare. So ranging from stress, pain, to inflammation and reduced survival rate. And this poor animal welfare is not only bad for the animal, but also invasive practices might lead to public outrage. Furthermore, many legislations worldwide require that impact on animal welfare is kept to a minimum. And stress and pain can affect behavior, so the results of our research might be misleading. So in conclusion, good animal welfare is not only important for the animals, but for the researchers as well. And one of the most commonly used guidance to safeguard animal welfare in research are the so-called 3 R's principles: replacement, reduction, refinement. And even though these principles were designed originally for research on lab animals, they can and they should be applied also in other disciplines of life sciences, including wildlife research. And the most obvious way to do that is to use non-invasive research methods instead of invasive ones. So going back to the research methods examples, if we want to collect DNA, we can just use something that the animals leave behind. If we want to identify individual animals, we can do that through natural markings. And to detect species, environmental DNA or camera traps work very well. Now the question is, are these non-invasive methods actually being used? And I recently conducted a survey among ecologists that work with wild animals, and only 39% knew the 3 R's principles, and only 22% used non-invasive research method in their last study. So you might be asking, like, why are these numbers so low? And it could be because there has not been enough guidance and resources specifically for wildlife researchers. So to help raise awareness, I created an online resource. So please have a look at it and recommend it to your colleagues that work on wild animals. And if you want to know more, then please come to talk to me to my poster or get in touch through an email. Thank you very much for your attention.
Thank you, Miriam. Okay, so finally in this session we have Mechtilde Wiegard from Berlin talking about training of animals as an applied tool for stress reduction.
So thanks for having the opportunity to talk here, and my topic is training and stress reduction in the context of refinement. Since we are obliged to develop continuously more gentle procedures when we use animals in studies, the blood sampling is mostly assessed as being a mild procedure. So we thought this might not be perfectly true for pigs. Since we take blood from the inferior vena cava or the jugular vein, the animals have to be fixed by a steel-wired sling, and this leads to retraction of the animal. They stretch their head, that's nice for us, but it's also combined with a lot of screaming, and it seems to be exhausting for the animals as well as for the humans. So we wanted to find out which extent of stress is really done by this procedure. So our questions were, how stressful is blood sampling from the jugular vein or the vena cava for a pig fixed with a sling? To which extent does either fixation or needle insertion contribute to the amount of stress? Can pigs be trained reliably so that fixation by an upper jaw sling can be omitted during blood collection? And can the stress of blood collection in pigs be reduced by training? So since you are only a person with an opinion, if you have no data, we set out to collect some of them. And we performed the first blood collection in the usual way, taking blood samples, determining serum cortisol. We took saliva cortisol samples before the sampling, 10, 30, and 50 minutes afterwards, and determined the heart frequency. Then we had a training period of 3 weeks, and afterwards we tried or performed the blood sampling in the trained way, as you see in the video, hopefully. We see here a mini pig trained to put the nose in this apparatus, being rewarded, and me taking the blood sample. And afterwards you see that the animal even turns to the sample taker to get some petting. Yeah. So here we see the values of the serum cortisol levels. We see that it was less in the trained animals, but we also did not find any significance due to the huge variation in the untrained animals. But when you have a look into the data of the individual animals, you see that the most excited animals, or the animals with the highest serum cortisol levels, benefited most from the training. When we have a look at the saliva cortisols, we also see that the untrained animals in red have a much higher cortisol level after blood sampling. The heart frequency was higher during the sampling in the trained as well as the untrained animals. Although if we have a look into the cause of it, you see that the baseline level was higher in the trained animals, as they were expecting a reward for the training. This is consolidated by other researchers. And you see a high increase in the untrained animals. So our conclusions are: blood sampling, including fixation of mini pigs in a sling, causes stress to the animals. The main contributor to stress is fixation. Mini pigs can be trained to tolerate aversive stimuli like the puncture of a cannula without fixation. Due to training, fixation-related stress can be reduced. This is implemented refinement, and we feel encouraged to do further projects on stress reduction through training, even in various species. So thanks to all involved. I appreciate the cooperation of the mini pigs, and thanks for your attention.
So, thank you to all of you. I wouldn't fancy trying to cram my research into 4 minutes, so thank you for doing an amazing job of keeping to time. We'll take questions for any of those 4 talks. Can you make sure you say who you've got a question for, if you've got one, please? Yep, go for it. Yep, whichever you like.
Hello, excellent. My question is for Miriam on the 3 Rs, and it's, do you know if there's a difference in the type of animals which are collected through invasive methods compared to non-invasive? Because if you're trying to get an animal, say moths, you can just use a light trap, which is an incredibly non-invasive method, but for other animals, is there a difference? Thank you.
Sorry, we didn't warn you.
I'm so sorry. I don't think I got the beginning of the question because sorry, I was running down.
It was basically just in your data, did you notice there was a difference between species? Because for some species, say moths, it's incredibly easy to get them using non-invasive methods like light trapping. But for other species, obviously, the type of method you're going to use can vary a lot. So did you see any difference within the species reported? Thank you.
Yeah, so my point is basically that we should use non-invasive methods whenever possible. It may not be possible in all circumstances with all species, but I think it's astounding that when we have the methods available, that they are still not being implemented. So my aim is actually like to raise awareness, to be more creative as a researcher, and coming up with non-invasive methods. So there are species differences, but I can't talk about the specifics. Sorry, I can't.
Thanks. There's one up there, and then Bas next, if you don't mind.
Thank you. I've got a question for, well, I've got two quick questions for Jasmine, if that's okay. So my first question is, you compared your decompression to CO2. I was wondering how you think it might compare to other methods like cervical dislocation, and whether that might be, I mean, if the person is trained, whether that's a better method for welfare. And then second quick question is, I wonder whether you think we should be thinking more about rehoming lab animals where possible too, so that we just avoid the issue of having to cull animals at all.
Okay, no, they're really good questions. First question with regards to comparing it to other methods. I think it's tricky because we were wanting to develop a high throughput alternative to CO2, because that's the only one at the moment that is available for people. Whether cervical dislocation and decapitation, if done properly, would be better is still kind of up for debate. They're obviously far faster. But it really does depend on how confident the person is that's performing that, and a lot of people, unfortunately, it's something that they don't want to face. So I think for us the priority was to find a high throughput alternative. As for your second question, absolutely it would be lovely. I think the trouble with a lot of mice is the genetic manipulation issue. I don't think it's really ethical to kind of maybe rehome them. And obviously, for some disease models, for example, I don't think prolonging suffering would be an ethical decision. But for more kind of wild-type mice, I think it should be kind of something that should be promoted. Thanks.
Thanks, Jasmine. Bas.
And a question for Holly, because I remember that you try to collect data from different countries in Europe. So I was just curious, did you see any variation between countries, or was the data set not sufficiently large for that? Or because it could be an opportunity for also countries and farmers to learn from each other, maybe?
Yeah, thank you. So we did try to collect a sample size from a variation of countries. It very much had more answers from the USA, the UK, very much the predominant sample, but that's kind of addressed in the first part of the paper, which looks at quantifying the rearing methods, and we did find some differences, particularly when it came to weaning ages and weights. That's quite variable. New Zealand in particular seemed to wean a lot later, and that's been really interesting when I've had conversations with them about gradual weaning, because they don't necessarily see the need, and I think that's because they're weaning later, so the rumens are developed more anyway. So there are differences, but it's in the first part of the survey, which is in a different paper, so we divided it into two, but they're both now published, so you can have a look. Thanks.
Thanks, Holly. I might have one for Mechtild if there's no one out there with other questions, which was just how much do you anticipate sort of resistance to using those kind of training methods, because they're quite time consuming? Is that the barrier to their uptake?
That's one of the barriers, but I think the time you spend at the beginning of training, you may serve while performing a study, especially if you have repeated interventions to do. So I think you should calculate for the whole, not only for the training phase.
Thank you. I can imagine that's difficult to do in practice, but I wish you the best of luck. Thanks you very much. Thank you very much for all of the speakers. Thank you, that was wonderful, and thank you for being so concise. So I'm just going to steal a bit of your time now to talk to you about UFAW's journal, Animal Welfare. If you're completely disinterested in academic publishing, I apologise for this, but we had a spare slot which allowed me to give you a bit of an update on our journal. So for those of you who don't know, as well as doing all the other things that we do, UFAW has published an academic journal, Animal Welfare, since 1992. We think it's probably the longest continuously published animal welfare journal. People had attempts at doing it before us, but all of those journals folded. And we've published a lot of really important animal welfare over those 30-odd years. I picked a fairly random sample here of controversial and really impactful studies, including Patrick Bateson's work on comparing culling and hunting deer, for instance. Lots of work on very controversial areas of humane slaughter and so on, right the way up to ducks in foie gras production. And the one that you heard about this morning, which was only just published, looking at the welfare impacts of rat control, which we think has had influence on policy in England at least, and hopefully the Scottish Government will be playing catch up soon as well. But there's a bit of a problem with our journal, and I've been acutely aware of this since I took over as both Editor-in-Chief and Chief Executive UFAW, and it's that we're publishing this journal using a very old-fashioned publishing model of being a closed access subscription journal. You pay money to get access to the articles, in other words, and that's a pretty unpopular publishing model in general. And it's really unsustainable in animal welfare, where we want the information that's published in our journal to have impact with the real world, not just in academia. We need this read by policymakers or people who can directly make changes based on what they read in animal welfare. So I've been very keen that we move away from that model. And also, publication animal welfare has often been quite slow, because the other thing is we publish 4 print editions per year, and if you get your paper accepted, it's quite possible you can wait, several months to actually get a space in one of our print editions, because we have a limited amount of space for papers. So all in all, we've been very keen to modernise how we publish animal welfare. And I'm really pleased to say that after a couple of years of myself and my joint editor-in-chief, Beate Nielsen, talking to lots of publishers and trying to find one who wasn't in it for the money, but was in it for the same reason that we're in it, we've managed to get a partnership with Cambridge University Press to turn the journal into a gold open access, continuously online publishing journal. We won't be printing copies and mailing them out anymore, so we'll be saving a few trees. And we hope we can publish much more rapidly, because once we've accepted your paper, assuming you as an author make all your proof corrections and so on in a timely manner, we can publish it in something like 22 days online. So I think it's a real step change for the way that we publish our journal. The one concern that usually arises when you move to an open access model is that someone has to pay somewhere, and if you're not charging the subscribers, then you're usually charging the authors to publish. And that's often problematic for people who don't necessarily have recourse to the funds to publish. But the good news is that there are various routes by which the vast majority of authors shouldn't directly be paying article processing charges to publish in animal welfare. So first of all, most authors in academic institutions, and we think more than 60% of our authors, will be covered by something called a transformative agreement, which used to be called a read and publish deal, which means that your university or institution has a deal where they have an arrangement with Cambridge University Press, which means that you just automatically don't have to pay your APCs because the university has effectively paid them in advance, and you can check your eligibility using those links. And as you can see, there are these kind of transformative agreements all around the world, and they are increasing as we speak. More importantly, I think, if authors don't have grant funds or any other recourse to funds to pay article processing charges, there's a very generous waiver system in place, which means that basically you just write to Cambridge University Press and tell them that you don't have recourse to funds, and you can be given a waiver for the entire article processing charge. And CUP deal with that, not UFAW, so the people who are deciding whether or not to accept your paper will not know whether or not it's had a waiver, which is really important for the independence of the editorial process. And finally, there are blanket waivers in place for the countries with the lowest GDP and various other measures of economic development in the world using a World Health Organization metric. So all those blue countries, nobody pays any article processing charges no matter what, and the yellowy gold coloured countries by default get at least 50% reduction. So all in all, we think we've got most of the bases covered in ensuring that there's access both to the information that we publish and allowing people to publish it in animal welfare. So just for those of you who are really interested in this and the timeline of our transition, the transition will actually formally take place on the 1st of January next year. Lots of the papers that are currently under review will probably end up being published in our new journal, as it were. And for about the next month or so, we won't be charging the article processing charges. So if you want to submit a paper really fast and have it published online next year, you can have it for free, even if you're one of those few people who have to pay the article processing charges. But after that, it will switch to the new model. So if you'll permit me to do a bit of advertising about why you should publish your research in animal welfare, or at least consider it. One thing is we actually have a pretty decent impact factor for the field, and it's been growing systematically over the last few years, and we think it will continue to grow. I know measures of impact like impact factor and site score aren't necessarily the most valid measures of the quality of a journal, but lots of people's heads of department or people who do their research impact probably care about it, even if you don't. So that's one thing. But I think the really important thing is the editorial service you get from animal welfare in terms of the integrity of the review process, which we hope is really thorough, and we will be striving to make sure is even more thorough in future, and the expertise of our editorial office. So if I just introduce you to the people who actually run this journal, obviously Beata and I are editors-in-chief, but I think the really important part of our process is that the people who steer your paper through to publication, even after acceptance, the managing editor and our sub-editor are both veterinary surgeons. One of them's got a PhD in animal welfare science. The people who are helping you get these papers ready for publication know about the science you're trying to publish. They're not just some random people in an editorial office somewhere, and that really helps us get papers of really high quality published in the journal. The names that are underlined are people who are in the room, by the way. So the managing editor, Steve, is over there, down there. Beata's up there somewhere in the middle, and Katie from Cambridge University Press is down here at the front. She's a life sciences publisher as well, so she really shares our vision for the journal. Feel free to speak to all of them if you'd like to about anything that we've been talking about. And this is our expert editorial board, and again there's a few names underlined there who should be in the room somewhere. So Jess Martin, who's from Edinburgh, edits the section on humane killing and slaughter. Bas, you've just heard from, and Sandra, you heard from earlier, who edits the section on wild animals. So we've got a really good editorial board. We're likely to want to grow that after this transition, so some of you might be getting emails asking you to join the board as well. But we think all of that offers you a really good process with animal welfare. So that's really it. I just wanted to give you that very quick update. I'm happy to answer questions if anyone has technical ones about publishing in animal welfare, but we'd be more than happy to talk to you one-to-one later on as well. But I hope some of you will consider publishing or keeping publishing with animal welfare, because we think it's a really, really positive change that all the information we publish is accessible to everyone now. And with that, unless anyone has any burning questions, which I'm not seeing, I will let you go and get lunch, which is the really important thing. So we're heading off for lunch. Don't forget the poster sessions. Lunch is upstairs and round the back as well. And please come back at 2:20. Yep. Thank you very much, everyone.
If you are presenting...